Okay, we're having this webinar for 2 days or 3 hours each. And the topic for the webinar is enhancing vaccine efficacy and unlocking the power of adjuvants in global vaccine development. Next slide. A little bit background about the scope and why we're doing this. The scope of this webinar is basically, it's educational in one way, and another way, it's also kind of providing a knowledge base for LMIC manufacturers about the vaccine adjuvants beyond aluminum. We're not discussing aluminum really today because we did that in 2024. And so we want to talk about adjuvants beyond aluminum, understanding the immunological principles, mechanism of action, regulatory consideration, all the manufacturing challenges, and strategies how we can improve vaccine efficacy and access. The participants, we hope to gain insights into understanding the mechanism of action and rationale for how we can select adjuvants and optimize them so that we can support local manufacturing in there and the CMC processes, and obviously foster innovation with all these new adjuvants that are coming up. A little bit about PATH. We are a global health organization and we are located in about 70 countries around the world. Our goal is to make health easier and provide access to low resource settings and affordable health products. And obviously vaccines is one of the big items that we look at as vaccines, and obviously strengthening the health services and advancing health policies. Um, in at PATH, we have a sustainable vaccine manufacturing program, which is supported by the Gates Foundation. And the goal here is to provide technical assistance to LMIC manufacturers to enhance their abilities to support them at various levels to ensure we can provide safe, effective, and affordable vaccines which meet international qualities and the public health program needs. On the next slide. Can we move to the next slide, Hannah? The logistics for today are, I'll go back. Okay. The logistics for today is during the webinar, all the participants will be muted excepting the speaker. And the webinar is being recorded and it will be shared on a PATH website in June. If you have any questions, please use the chat feature. These questions will be monitored. We'll collect them and we'll either be able to take the questions after the speaker or at the end of the webinar today, depending upon the timing and how the opportunity presents itself. After the webinar, we will send out a brief survey to all of you to get some feedback on webinar, because we want to understand how this is impacting and is helpful so that we can bring more of these kind of webinars out on through PATH. On the next slide, today's agenda is a brief introduction that I've already given. And let me introduce myself at this point now. My name is Lakshmi Khanke. I'm a Senior Program Advisor at PATH and hosting this webinar. Today's speakers are, first speaker is Derek O'Hagan from GSK. After that, we have Dr. Julie Joseph from Health Canada, followed by Mark Alderson. The questions and answers and the moderator for that will be Robert Joy from PATH, which will be after the three speakers. So a little bit before we get Dr. Derek O'Hagan, a little biography on him. Actually, Derek O'Hagan doesn't require any introduction in the field of adjuvants. He's so well known for years and years. We all know him as one of the pioneers in new adjuvants, and he's right now the global head of discovery support and new technology at GSK within the R&D, and he has a number of publications. And if you Google vaccines, you will see so many publications coming from Derek. He has multiple patents under his name and even several books on adjuvants. So with that introduction, I'd like to invite Dr. Derek O'Hagan for his talk. Derek, you could take it on.
Yeah. Sure. I'm just you're going to run the slide, so I just sit here and chat away. That's the plan.
Yes.
Okay.
Hannah will do that. Hannah will.
Okay. So good morning, or whatever time of day it is over where you are. So I'm going to talk about vaccine adjuvants, and I'm going to contradict my chairperson already. I will talk about alum because I believe it's the future, not just the past. But I'll talk about a lot of other adjuvants too. So maybe let's go into the first one. First slide, sorry, change slide. So probably this is not a surprise to anyone. Adjuvants are not easy to define for a variety of reasons, but probably this is the best definition. You add them to vaccine antigens and they enhance the immune response to the co-administered antigen. And there are many, many different adjuvants. You'll probably hear about lots today and tomorrow. Importantly, or confusingly, adjuvants is a term used in other settings, but I will use it in this setting, something administered in conjunction with the vaccine antigen that results in an enhanced immune response to the antigen, and it can manifest in a variety of ways, and you see here. So that's what, when I talk of vaccine adjuvants, that's what I think of. And that's what I'll be talking about exclusively. And maybe some of the other speakers, if they're talking about using adjuvants in different ways or want to define them differently, then please, maybe they can do that to hopefully not add confusion. So next slide, please. And probably I need to update this at some point. But in essence, it's the big picture. Yes, we have had vaccine adjuvants for quite some time, about 90 years. And of course, alum was the first and is still used, very safe and effective, has limitations in performance. And that's how and why the others have emerged. It was slow, challenging, difficult for a long, long time. Some people would argue it still is to get novel adjuvants out there. I would be one of the people who would argue, yes, it is. It is quite challenging. Nevertheless, it has been accomplished. There have been several more in the recent past. You see the kind of orange color, that's the GSK adjuvant systems. You see MF59, which was arguably one that changed things. And there are many kind of almost generics copies of MF59 that have emerged subsequently, relatively recently. CPG and Ischematrix have emerged, and of course the pandemic opened up some opportunities. But yeah, we've had vaccine adjuvants for quite some time. Mostly it was alum based. Now some novel technologies are starting to emerge, and we'll talk about why as I go through the talk. Next, please. And so this is something I'm trying to remember the date. Probably, 12 years ago, no, 13 years ago, I kind of launched this out as what I thought was a rational approach to R&D around vaccine adjuvants. And I look at it regularly because I do talks like this, and I think, probably pretty right. Maybe some things could be modified. But pretty much you start with, well, if you don't need them, don't use them. And of course, there's a variety of ways you can, within the realm of antigen design, where we can engineer antigens as VLPs, optimal representation of epitopes. So we can make pretty good antigens. So if you can use just the antigen, that's simpler. Make a vaccine with the antigen. If you need an adjuvant, then thoroughly evaluate alum, and I'll talk about how to do that. But it's safe, effective, well established, inexpensive, broadly available. It's kind of the go-to, the starting point, and really it should be the benchmark, because unless whatever you're going to use significantly outperforms it, then maybe just stick with alum. And generally speaking, oil and water emulsions can be a better option. They're likely more potent. But MF59 was probably was the first that was included in licensed products in flu vaccines. Now there's much more options available. Some very similar, some different. So oil and water emulsions are a good option. Not so good on CMC matters. I'll maybe talk about that. But yeah, there are more novel approaches, and we'll talk about them next couple of days. I'll introduce some today. Whether you need them, yeah, it depends on your population, depends on the vaccine target. It depends on whether people have been pre-exposed or they're naive, lots of factors. But generally speaking, if you need something more potent than the very established ones, you're going to need to work on establishing mechanism of action. You're going to need more data, longer timelines, more regulatory scrutiny. If it's novel, if it's different, if it's not been seen before, inevitably there's going to be a lot more challenge associated with it. I think we all understand that. And the idea of, well, alum is today and the future, not just the past. I'll talk about alum plus, generally TLR agonist adsorbed onto alum, which I think is an emerging popular approach. So yeah, alum is not going away, and nor should it. And then, yeah, probably final point. Yeah, don't think that those adjuvants developed for injection are going to be effective for alternative routes. Generally not at all for mucosal delivery. So happy to debate these points. I think they're still fairly rational. But maybe we won't have a debate today, but at some point in the future. So next slide, please. And here's my alum one. And the message here is really the importance of formulation science. Maybe I'm a little biased because I'm a formulation scientist, but in essence, optimizing alum and its use to really determine if it can do the job for you before you take adventures with more complex adjuvants or adjuvants that have bigger challenges on CMC side. It's a good idea to try and utilize alum. Obviously, the industry has great experience here, but so do many academic scientists around the world. Generally, formulation scientists. Tom Hogan-Esch is a guy I wrote this with. He's not actually a formulation scientist. He's a veterinarian, but he knows alum very well. So yeah, I just encourage people. There's lots of factors in and around the alum and the antigen and the buffer system, excipients of how you optimize the use. So please, this is a review that gives a high level, what are the factors to consider. There's lots of literature and there's lots of experts who can help you and getting the best out of this well-established adjuvant. Next, please. And sorry, this is kind of an example of the attributes, the positive features that alum brings. And I chose this one of my papers because it was readily accessible. But in essence, adsorption to alum can stabilize the vaccine antigen and can enable your preferred product presentation, which is always pre-filled syringe or a vial that can be liquid, that can be stable as a liquid in a fridge, etc. So here you just see these are the recombinant antigens included in Bexsero. So you can see here that the adsorption to alum enhances thermal stability, maintains antigen recognition by antibodies, and inhibits antigen aggregation. So this is an example where 3 recombinant proteins were stabilized by alum adsorption, and that enabled a liquid, fridge-stable vaccine formulation. And that's an example that you can see in many settings. So alum has pretty attractive features from a CMC perspective, and we shouldn't lose sight of that. Next, please. And I mentioned MF59 earlier. MF59 is an oil in water squalene emulsion. I feel qualified to talk about it since I worked on its R&D for 20 years, quite some time ago now. And this is illuminating what I said earlier. Emulsions often can be more potent than alum. So this is, in fact, a phase 3 flu efficacy trial in young children. And, you don't need to be a sophisticated statistician to see the difference here. You've got TIV, which at the time was trivalent influenza vaccine. That's the vaccine efficacy in purple. And then you've got Fluad, which is the same vaccine with MF59 oil and water emulsion. adjuvant, and then you see the enhancement in efficacy, and you see barely any overlap between the two curves. Young children, which are a population for which adjuvants can be quite effective, because they've not really been exposed much to flu yet. So this was an example of phase 3 efficacy. You see a significant enhancement in efficacy. I think we all know alum does not work well for these kind of influenza vaccines. Emulsions work very well. But to the right, you see, acceptable safety profile, but you see increased local reactogenicity, no increase in serious adverse effects, but you do see more systemic reactions. So the balance is always there with adjuvants. You get something, enhanced responses. Let's hope those enhanced responses are necessary and can enable the product, and you couldn't get the product without them. But you're going to see increased local reactions, and you're going to see probably some increased systemic reactions. So win some, you lose some. This is the reality. So next, please. And this is an important paper, and I always say that one is one of mine. That's just the way I work. But I like this one because, in essence, it was starting to change the perspective. So this was utilizing MF59. We got it licensed in Europe, and then we were trying to get it licensed in the US. And in essence, what we understood was we needed a story of how it did what it did, its mechanism of action. And that's what the US regulatory agency was demanding at the time. And there'd been a lot of kind of hand waving of how do adjuvants work, what they do. And okay, well, alum is kind of controlled release, it's a depot. And then our emulsions, well, oil and water emulsions aren't. So what do they do? And this was a study, I guess it's 17 years ago, utilizing the technology at the time. And it was looking at gene expression profiling in the muscle and the local lymph node. And the key message is MF59 was acting as a local activator of innate immunity. So this is muscle, and these are kind of immune-associated genes, cytokine activity, cytokine binding. You see the controlled injection of saline, then you see in the middle MF59 lighting up lots of gene modules, and then you see alum to the right, much less. And then to the right, you see the Venn diagram, a lot of overlap, poor feature of different adjuvants, but you see a lot more local activation with MF59. And, CPG was included in this. And surprisingly, we saw a lot more local activation with MF59 than CPG, which was described as an innate activator. So it was interesting. There was a story developed of recruitment activation of immune cells, often neutrophils, in fact, and, then moving off to the local lymph node. But this was an important paper because it started to define how this class of adjuvants, oil and water emulsions, really worked. And it wasn't what people have been talking about for a few years. And it certainly wasn't depot. I'm frustrated every time I review a paper on emulsions, oil and water, and people tell me it's a depot. Never was, never is. Next, please. And this is kind of expanding the story and bringing it more up to date. This is a review from 5 years ago, and it was talking about the different emulsion adjuvants. It was talking about what we've learned about the mechanism of action, obviously including the signals I talked about in the previous paper and many subsequent papers. We did a lot of work on MF59, and other emulsion adjuvants were also evaluated. To the bottom right, you see that the outcome: increased T cell, long-lived plasma cells, expanded repertoire, etc. And then to the left, you kind of see, trying to see what I mentioned of recruitment to the local muscle, activation of those immune cells, picking up antigen, taking them off to the local lymph node, along with the activation signals, and that's where the immune response is induced. So we know quite a bit now about how oil and water emulsions adjuvants work in particular. Next one, please. And yeah, and maybe talking about oil and water emulsion adjuvants, maybe I need to separate. So, of course, GSK has one which is called ASO3. It differs from the others because pretty much MF59 and most of the others that have emerged that are similar are all squalene only. ASO3 is squalene and vitamin E, which is, if you use it at a very low dose, it's an antioxidant, and some of the emulsions have it as an antioxidant to stabilize the squalene, but it's actually a standalone adjuvant in veterinary vaccines. And then you look to the right here and you see a study that showed you squalene only, which is essentially ASO3 without tocopherol, and then ASO3. That's just a mouse study on the right, but you see the clear differentiation as a result of the inclusion of the alpha tocopherol, and you see the structure to the left. And now there's a number of studies looking at different potential pandemic flu vaccines where head-to-head ASO3 and MF59 have been evaluated, and yet ASO3 tends to be more potent than the squalene-only emulsion adjuvants. So next, please. Started with the kind of particulate alum MF59. I'll get onto liposomes eventually, but they were the kind of the first generation for which we kind of understood how they were working. And then this marvelous insight emerged in the Nobel Prize was awarded in 2011 to these distinguished gentlemen on the bottom. I think the first publication was about 20 years ago now. And essentially it was the identification of toll-like receptors recognizing microbial structures. And of course, this is a pathogen recognition system. These are pathogen recognition receptors. And you see a very simplified version of the downstream cascade that's triggered, activate innate immunity, and the recognition that this then drives adaptive immunity. And then essentially molecules that we've been using as adjuvants, and I highlight two here, which were subsequently successfully developed. MPL, which is a TLR4 agonist. TLR4 is an extracellular receptor, and CpG, TLR9 agonist. That's an intracellular endosomal receptor. So in essence, these molecules and many others and various extracts of bacteria and viruses, they were adjuvants, and for decades they'd been described as adjuvants, and they have an adjuvant effect. And again, lots of hand waving, and then here's the mechanistic interpretation of the receptor system that they activate and why they are successful adjuvants. So this was a crucial insight in the world of developing vaccine adjuvants. Next, please. And it certainly opened up big opportunities. And one of the big opportunities will show up on the next slide. And so once you've got a receptor, okay, so then you can start to do discovery. And of course, for large pharma companies, I was in Novartis at the time, they have discovery engines for small molecules. So now we know that we have receptors, TLRs. Now we can do discovery. And so the first one we focused on was TLR7, and the story was here. So we know TLR7 is expressed on human immune cells, type PDCs in particular, and you're going to get a type 1 interferon response. TLR7 is actually validated already as a human drug target, and the story is to your right. Imiquimod, it was called an immune response modifier. It was actually discovered and used for actinic keratosis and basal cell carcinoma before people understood its mechanism. It was meant to be a fast follower of acyclovir, and it turned out it was effective in these settings. They didn't know at the time because it was a TLR agonist. So TLR7 was a validated target. You see here, people had actually used it as a topical adjuvant, and then there were many other TLR7 agonists in development for different indications. So we focused a discovery effort on TLR7 as a target. And this is going back probably 15 years or so. Next, please. Was published about a decade or so ago. And we come up with a fun title. Well, Better Adjuvants Through Chemistry was the title, but in essence, this is the discovery of what we call small molecule immune potentiators. And I'll show you what I mean in a subsequent slide. But what we discovered and how we used it is on your left. But we had to come up with a pictorial representation, which was used in the paper on the right. And it was trying to illustrate what kind of characteristics and physicochemical parameters are necessary to succeed. And of course, I already highlighted, we already had SMIPs. So imiquimod and resiquimod were available, but they would result in the big negative no that you see with the kind of reddish person. They would have poor efficacy and poor tolerability, because in essence, they were soluble molecules that if you used as vaccine adjuvants, the way I described, you inject them with the vaccine antigen, they would rapidly diffuse away. So they're easy to formulate. You can just put them in the vaccine, but they have a very short in vivo half-life. And the problem is that they are freely soluble, so they move away from the injection site, which would result in poor tolerability. So the drugs we were working on, we tried to exploit different characteristics of them, and we created what was called first generation SMIPs. And at the time, the immunologists were ready to declare victory because they had good efficacy and good tolerability, simply because we changed the solubility of the molecules. But that made them very difficult to formulate and deal with dose uniformity. And it gave them a very long in vivo half-life. So in essence, they were insoluble. So they didn't have the problem of the existing SMIPs going away, causing local systemic reactions. They stayed at the injection site, but they stayed for extended time periods. And of course, because they were insoluble, they weren't easy to formulate in the liquid formulations that we wanted. So with that chemotype, we modified the chemistry and got second generation SMIPs, which were better efficacy, good tolerability, short in vivo half-life, and easy to formulate. And if we move on, that's what I'll talk about. And it's all wrapped up in that paper from 12 years ago, if people are more curious. So next, please. So this is trying to differentiate why we thought small molecule immune potentiators were the future. And, you can look at the words, but look at the bottom of the structures. And that's really trying to illustrate it. On the right, you've got your TLR7 agonist small molecule immune potentiator, and benzonafthridine was the chemical structure. Simple, small molecule. And then you look at the others that I'll be talking about later as well. TLR4, NPL. Molecule from bacteria, a large complex molecule produced essentially through biotechnology, aggregates, difficult to formulate. Okay, and then you've got pan3cis, similar challenges. You've got saponins, some of the most complex structures of any drug-like molecule available. So the one to the right, the SMIP, was like, yeah, that's going to be easier. So we're creating simple synthetic molecules. Obviously, we're looking to minimize supply chain, production challenges, simple synthesis. They are discrete chemical identity, so no heterogeneity. You can control the level of agonism, and then the structure can be easily manipulated. And we manipulated it so it would adsorb to alum. But essentially, we added a phosphonate group, which would then ligand exchange bind to alum. Analytics are simple, and I guess we thought we've been working on these kind of drugs for a long time, so probably we figured out how to work with them. So we thought SMIPs were a good idea. Still do, actually. Next, please. And I kind of already covered this, but we created the molecules and we're like, okay, well, how do we deliver them? And we could use emulsions, we could use liposomes, we could use nanoparticles, and we did all those things because we like having fun with formulation science. But what's the best way to deliver them to take them into the clinic? And we thought, well, let's just absorb them to alum. And this is my story. Alum is the future, not just the past. So alum, well, why use it? Well, it's readily available, inexpensive, safe, well tolerated, known by the agencies, the degree of comfort with it. It allows co-delivery of the antigen and the SMIP if they're both co-absorbed. You can then use the drugs, the SMIPs, with existing vaccines because if they're already absorbed to alum, you can modify them, make them more potent. And in essence, this had been a path that had already been proven. So ASO4 is a TLR4 agonist, MPL, adsorbed to alum, which had already been included in licensed products. So the idea of absorbing a SMIP a TLR agonist to alum was already somewhat proven. And of course, like I already mentioned, this alum TLR7 SMIP absorption enables single dose vials, pre-filled syringe, fridge stable. These are the kind of products we want to utilize. So we call this AS37 once we arrived in GSK because it needed an adjuvant system number, and the story's at the bottom to the right. So yeah, AS37, I'll talk about a little bit more. Next, please. And this is one of my favorite studies. And it's one of my favorite studies because it really how one should biologically evaluate a new adjuvant. And what you do is benchmark it and you challenge it and you say, so this was the full story. Joe Francica was the lead person on this. He was a postdoc in the lab of NIH, of Bob Cedar at NIH. Bob had been in HIV research for a long, long time, had a number of connections with different collaborators. He challenged us and said, hey, if you think your neoadjuvant is good, why don't you try it for HIV and glycoprotein and try everything else and tell me if it beats it or not. And that's what we did. So this is non-human primates. It's like 6 per group. But I like it because you lay out the adjuvant world. You start with the protein only on your left. Then you absorb the protein to alum, and you see some benefit of alum. And then you say, okay, well, TLR4 agonists are readily available. Let's see if they're any good. And yeah, they make it better. You see in the green. And then you say, okay, well, here's TLR7 on alum AS37. How does that do? And it does okay. It's better than the TLR4. That's good. But you've got to benchmark it too. If you're just looking for neutralizing antibodies, well, MF59 does pretty well. And then TLR4 and TLR7 added to an emulsion didn't seem to add any benefit. Maybe that says the emulsions are pretty good anyway. And then to the right, you've got Matrix, which is now Matrix M. Yeah, pretty good adjuvant. And you've got poly-ICLC, which Bob considered his positive control. I told him he wouldn't get that licensed ever. Maybe it could be a debate, but he believed that it was his positive control. So you see across the profile, adjuvants, what they do, some are better than others, but you need to understand how well yours is doing. And there's the neutralizing antibody response in non-human primates. Next one, please. But of course, neutralizing antibodies is interesting and important, but you can look at other functionalities. And this is with Guido Ferrari, one of Bob's collaborators, and he looked at ADCC, which was thought to be very important. And then you see again, the protein doesn't do much. Alum helps. I'm not sure TLR4 helps here, but TLR7 helps quite a bit. And even with this small group of 6 non-human primates, you saw a statistically significant response of alum TLR7 over alum alone. And then you saw that MF59 was pretty good, but obviously a much broader spread. And then you see the other kind of comparators and controls to the right. Next, please. And again, not doing the full profile, but of course, the T cell response was of interest. This is the ELISpot response, and it's all in this paper and another subsequent paper that Joe put out, again, almost a decade ago. And you see the groups again here. I'm highlighting the AS37 as the one that's in pink. And in essence, it's up there with what Bob considered really positive control, poly-ICLC, and also Matrix, which is known to give a good Th1 cellular immune response. The others tending to be much weaker, including MF59. And this is where you see some of the limitations. Emulsions tend to be more potent than alum, but they're not particularly potent at trying to give you a Th1 response. That's where you need an additional or a different signal, and perhaps TLR7 activation is one of those key signals. So next, please. So there's a lot of, in fact, more than one study, but it's an important signal, an important message, and it's showing consistency between non-human primates on the left and humans on the right. So we published this a few years ago now. So on the left is a summary of the same study you just saw, evaluating the different adjuvants. And we're looking at the transcriptomics response in these non-human primates. And we've obviously, instead of all the groups, we've made it just 3 groups because we want to make a comparison. And what you see are the, which genes were upregulated in these non-human primates. You see not a lot of activation. And this is 24 hours after immunization in blood. Not much activation with alum. And then you see alum TLR4 is the green one. You see TLR4 is activating an inflammatory modules. Okay, that's interesting. And then you see the pink one, which is AS37, alum TLR7. So you see it's activating a completely different set of genes associated with interferon. And that's exactly what you'd predict from TLR7 present on PTCs. So this and a lot of preclinical studies kind of told us, yeah, the TLR7 is doing exactly what we want it to do, and it's doing something different from TLR4. Because if it was the same as TLR4, well, why bother with it? Because you've already got that. So we wanted something different. That's what we were getting. You saw the adaptive responses were different in the previous slide, and here you're seeing that the gene modules activated in peripheral blood were different too. And then to the right, you see the first human study with AS37. This was with a conjugate vaccine, not because conjugate vaccines are a good way of showing an adjuvant effect, because they're very safe, very well tolerated, and they already have alum. So then, in essence, what you're looking at is the safety tolerability of the TLR7 agonist only. But in essence, the message here is exactly the same gene modules were activated in the non-human primate to the left and in the human to the right. So in essence, the molecule was doing what we hoped it would do in a very specific fashion. And again, this is published if people want to look at it more closely. So next, please. Okay, so I talked about we like benchmarking, and I'll talk about that again later. But here's the benchmarking of the new adjuvant AS37. And what we've done before is evaluated hepatitis B surface antigen with other adjuvant systems, and I'll show a bit of that later. And here's a trial recently completed where we did the same. So we're trying to benchmark, is AS37 a good adjuvant relative to some of the others? So we have the baseline alum. We have a comparator, which is ASO4, so a TLR4 agonist, which is that one's included in licensed products. So it's a good benchmark for safety and potency. And then we put an emulsion in there, ASO3, or a squalene oil and water emulsion with tocopherol that I mentioned earlier, because emulsions tend to be very potent. And so alum, the product needs 3 doses, and we decided to try the other ones with 2 doses only. So the regimen is 31 days. The alum was boosted, and we tried AS37 at 2 dose levels. These 2 dose levels had been evaluated in the previous trial, and it wasn't clear which was the best dose level. So I'll run through this data quickly. Next, please. So first of all, you've got to consider the reactogenicity. So this was administration site adverse events, and pretty much only pain was reported. And then you look across the groups. Not surprisingly, alum is better tolerated than anything else. And all of them were kind of better on second dose than first dose. Not really a lot of differences other than maybe a few grade 2 reactions in ASO4 and ASO3. Next, please. This is looking at systemic reactogenicity. And now you see all adjuvants give some systemic reactions. They were all mild to moderate, a couple of days duration. And you see pretty much alum also gives local reactions. You see generally better tolerated second dose, and they're all kind of about the same. Maybe on second dose, you could argue that ASO4 had more reactions than alum, and maybe AS37 was more similar to alum. But these are relatively this is like 25 individuals. But yeah, reactogenicity seemed okay, and AS37 seemed to be somewhat similar to alum. Next, please. But you certainly saw significant differences in the immune response. So that the curves are on top. So day 31 is after a single dose, and then you see the color scheme tells you which is which. The purple is the highest dose of AS37, and the blue, which is standing out, is ASO3. So after one dose, you see AS37 and ASO3 given good responses. After two doses, day 61, you see AS37 at the lower dose also given good responses. And you can look at it different ways. You can look at the curves and the colors, or you could see the seroconversion rate, which is the table in the middle. And what's quite striking, alum takes the 3 doses to get to seroconversion, remembering that those subjects had a third dose. The other groups all had 2 doses only. You see very high seroconversion with ASO3 after a single dose. You see similar high levels of seroconversion with AS37, 50 and 100 micrograms after a single dose, complete seroconversion after 2 doses. And then you see ASO4 low seroconversion after 1 dose and kind of catching up after 2. So take your own messages here. Clearly, AS37, ASO3 were the most potent, gave seroconversion after a single dose, outperforming alum for sure, generally outperforming ASO4. So good to see the benchmarks and where the new one sits. Next, please. A little bit more of the data. This is looking at the frequency of hepatitis B specific memory B cells, and you pretty much see a similar picture. You see ASO3, which is a very potent and safe adjuvant, doing a good job. You see AS37 at 50 and 100 micrograms, also doing well and outperforming its benchmarks of alum only and ASO4. So AS37 looking good relative to its alum-based counterparts and doing its best to catch up with ASO3. Next, please. And so I've talked for quite a while without really diving into the established adjuvant systems, but you kind of heard me mention them. And this is just highlighting what they actually are, if it wasn't clear already. Obviously, what we build are delivery systems, which can be emulsion, alum, or liposomes. And then we start to add immune potentiators, which vary. ASO4, ASO3, ASO1 are all included in licensed products. And I mentioned AS37 with the TLR7 small molecule immune potentiator in early stages of clinical development, but given a kind of an attractive and interesting profile. So next, please. And I mentioned early that we do this quite commonly. So we try to benchmark the adjuvant systems to try and understand which one can do the job that we're looking for in a particular pathogen-based vaccine. And this is an earlier version of the study you just saw, but of course not with AS37 because it wasn't available. So this is again hepatitis B surface antigen publication from about a decade ago, and it's comparing adjuvant system 01 at different dose levels with 03, 04, and alum. So like I said, this is a clinical study, and this is just high level message. If you look at the top, you're looking at the level of T cell response, and you see AS01 is the most potent by far. AS03 does well. AS04 somewhat better than alum, but not so good. Bottom left, you see the antibody titers with AS01, AS03 all doing well. AS04 doing better than alum. And then to the bottom right, you see the downside. You're always going to see more local reactions. And then, again, we're always benchmarking with alum. And then you see the local reactions, they're all more local reactions than alum. And then you see the systemic reactions, and yes, they're all more more local and systemic than alum only. So this is the profile of adjuvants. Generally speaking, you get more local and systemic reactions, but that's countered with more enhanced responses, T cell, B cell, and functional antibodies. But if you want to understand where your adjuvant sits, this kind of study and benchmarking is a good way to go. So next, please. And this is a kind of commercial slide. I kind of couldn't avoid because I want to talk about AS01, and I can't talk about it without highlighting in where it's been used. So it was included in the first malaria vaccine that is now shown to do a great job in preventing children's deaths. It's one of 2 approved malaria vaccines now. But it's also worked well in Shingrix, and then you see that's the top level message. Regardless of the age of the individual, there's a remarkably high degree of protection against shingles, which is sustained over more than a decade. And then below, more recently, Arexvy is the product. It was shown to be a highly efficacious vaccine against RSV. Both these vaccines are utilized in the elderly. Both are licensed in a number of countries, US and Europe, and all this data was published. But in essence, I wanted to use this to illustrate AS01, safe, effective adjuvant, doing a great job, particularly in the elderly with these 2 products, but also enabling the first malaria vaccine. And I want to talk a little bit more about AS01. So next, please. So this is the composition of AS01, and it's the most complex adjuvant included currently in a licensed product. It has 2 immune potentiators, both are natural products. Bottom left, I'd already talked to you about NPL, a TLR4 agonist, and then bottom central is QS21, which is a saponin, and has a remarkably complex structure, as you will see there. Both are natural products, so have significant heterogeneity. And both of these molecules are incorporated into a liposome with DOPC and cholesterol. So it's a multi-component adjuvant system. Next, please. But even from the first studies, it was very clear that all these components were necessary to get potent T cells and antibody responses, but also that there was real synergy that emerged as a consequence of the signaling of both NPL and QS21 together being driven into the local lymph node by the liposome that's incorporated in. And you see that down at the bottom here. This is a mouse study from 14 years ago, and it's illustrating everything can be added and removed. Liposomes, not really a very effective adjuvant. NPL and liposomes, a little bit better. QS21 in liposomes, yeah, a little bit better, but you're not really getting a great response. The only way you get the great antibody T cell and polyfunctionality of the T cells is the MPL and the QS21 in the liposome. So it's the formulation of those elements together, ensuring delivery into the lymph node that results in the key immune response. And like I said, this was a response against GE in varicella zoster that went into the Shingrix product. So next, please. And so, yeah, I talked about mechanism of action being crucial to the path to licensure in the US in particular. And certainly AS01 has a complex mechanism of action because it has two immune potentiators. So it's triggering known receptors and target cells. There's local activation at the injection site, but then the liposomes and the activation signals move to the local lymph node, and there's a lot of activation in the local lymph node. And this is a kind of summary of what you're seeing, of what TLR4 does and the impact of QS21 on the inflammasome. And there's a bunch of papers that describe the mechanism of action in all its complexity. But that's, again, illustrating the point. The path to licensure is through mechanistic understanding of what the adjuvant does. And this is all about the transient innate response driven by the combination of these elements over the first couple of days. And a lot of this has been able to be translated into humans by doing complex systems biology studies, looking for the signals in humans and various other preclinical models. So next, please. So this is a common story, and it's not an easy story on supply chain. So in essence, Quilea saponaria is the tree from which QS21 comes from. It's one of many fractions. The tree grows in a limited geographical area. And tree bark is the source. So this is probably not your ideal supply chain, but like all large pharmaceutical companies, we develop an awful lot of diligence around the supply chain and what's necessary to make sure it's sustainable. And I'm going to talk a little bit about some of the steps there. So next slide, please. So this is looking a bit more closely at QS21. And it's described as a single fraction because it was described as a single fraction by Charlotte Kensel in 1992, I believe, I remember correctly. But of course, once the analytical capabilities improve, one can look more closely and discover that, in fact, it's not a single fraction, but multiple fractions together, both major and minor components. So not only is it complex, it's heterogeneous, and there are a number of molecules present. Next, please. And over to the left, you're starting to see the kind of complexity you see in a crude, this is UPLC of a crude bark extract. And you see, yeah, QS21 is there. It looks like a single fraction, it clearly isn't. And you also see other significant fractions. QS17 is similar in quantity to QS21, but QS18 is more abundant. So for years, we spent a lot of time purifying QS21, and the rest was kind of waste. And then we thought, well, maybe that's not the best approach, and maybe we could do something different if we wanted to improve the sustainability of the supply chain. And 18 and 17 differ from QS21 only in the presence of a few sugars and different sugars. So then we thought, well, maybe we could change those sugars. So next, please. And so this reflects the effort of some talented scientists within GSK. And again, it's part of the story of why being in a large pharma is often advantageous, because you've got a lot of scientists around you who have great capabilities to do interesting, exciting things. And here what they did was identify enzymes that removed the sugars that converted 17 and 18 to 21. Then they worked with those enzymes to make them much more potent and came up with enzymes that did a much better job. And then hopefully you can see it on your screen. You see the original profile of crude bark extract in red with QS18 and QS17 present. And then in the green line, you see the treated crude bark extract, which has been treated with these engineered enzymes. And in essence, 17 is gone, 18 is gone, and it's all been converted to QS21. So essentially, we exploited these enzymes to convert what was previously waste materials into what we wanted, which is QS21. So you see to the left, there was a significant increase, at least fivefold in QS21 after chromatography. So this is a step along the path to better sustainability of the supply chain for QS21. Next, please. And so this is a story I'm not going to dive into, but of course, it's a very interesting story, and it's not a GSK story. So these products incorporating AS01, both to protect against Shingrix and VZV and to protect against RXV, to protect against RSV or XV. Stories are starting to emerge, and this is all the story around the zoster vaccine, of additional benefits emerging. I think most of these relate to evidence from large population databases in different countries that the utilization of the recombinant zoster vaccine results in a significant reduction in incidence of dementia. And now we're seeing somewhat similar signals start to emerge in favorable outcomes from a cardiovascular perspective. And it's even, so then you might think, okay, so great, virus is bad, we protect against varicella zoster, vaccine is doing its job. Simple story, protect against the viruses, an additional benefit of vaccines. But now we're seeing RXV have similar signals start to emerge, particularly in the cardiovascular space. That's a completely different virus. So it's an interesting story. GSK don't own it, but it's good to see these signals emerging with these products being used. I think there will be a lot of interest in this story as we go forward. So next, please. So now I'm going to get back to what I should be talking about, I think, new adjuvants. And there's lots of opportunities. And this was from a review a couple of years ago where we were highlighting different TLRs that had been looked at extensively, that are all mostly being exploited. There's plenty of other receptors, whether it's the inflammasome, whether it's RIG-I, NDA5, whether it's STING, and I put a red box around STING, so I'm going to talk about that. They're all additional cytosolic targets of activating innate immunity. There's a bunch of C-type lectin receptors. Probably Jay will, and maybe Dennis will talk about MINCL at some point in this. So there's lots of other, so far, underexploited receptors. And then over to your left, as we learn more about how adjuvants actually work, there's lots of opportunities, controlled cell death, tissue damage, metabolic adjuvants, epigenetic adjuvants. So there's lots of potential targets and new mechanistic insights emerging. So I'll focus on just one, STING. And you see that to the right of the cytosolic targets on the top panel. So next, please. So yeah, this is summarizing stimulator of interferon genes as another, a receptor that recognizes infection. It's another innate activator. You see the signaling pathway through type 1 interferons to the left. You probably can't see very clearly, but the story to the right is a published story. GSK published in 2018 in Nature that we had discovered molecules that were synthetic sting agonists. So we are calling them diabzi, and you see at the bottom, the diabzi, they needed to dimerize the molecule, and then it was comparable to the natural product in terms of activation. So the discovery effort was done by GSK for a different indication. And now we're seeing, can we exploit these molecules to be vaccine adjuvants? So next, please. And this is a story you've heard already. Sting is another small molecule immune potentiator with the advantages we talked about earlier for TLR7 agonists. And the same debate, well, how best do we deliver them? Well, I said, well, let's do the same as we did with AS37. Let's modify the structures. Let's make them adsorb to alum. And then we can create the same single vials or pre-filled syringes. And we will accumulate the same positive attributes for the inclusion of alum that I highlighted earlier. So that's the story. We repeated the story. And so today I'm introducing the next generation AS34, which is a sting agonist adsorbed to alum. So next, please. So this is looking at validating whether what we tried to do worked again. And on the right is the pharmacokinetics, which is in essence the circulating level of the drug. And it's the same story I told you. It's freely soluble. If you inject it with your antigen, it would diffuse away from the injection site, and we think that would result in systemic adverse events. So we're adsorbing it to alum. So on the right, you see the PK profile. You see the drug on the top, injected in blue, or whatever you want to call that color. And then below, you see AS34. You see it adsorbed to alum. And what you can see is a 8 to 10-fold lower serum Cmax for the drug adsorbed to alum. So you've changed the PK, you've focused it on the injection site. And then you look at what the impact is, and you look to the left. And you're looking at different cytokines in the plasma. And we're actually benchmarking it with AS01. So on the top, you're seeing it does what it's meant to do. So you're looking at interferon alpha and interferon beta. And the little red dot is what AS01 does. So what you're seeing is, the sting is doing what you expect it to do. It's the stimulator of interferon. You're getting these alpha and beta interferons, type 1 interferons. And you're seeing that you've blunted. Again, the blue is the drug alone, and the green is the drug adsorbed to alum. And this is systemic cytokines. So, of course, the drug alone gives high systemic cytokines. You adsorb it to alum and you block that. And then below, you're looking at the characteristic features, perhaps, or some of them, of AS01. Again, to the right, gamma interferon is what you see with AS01. This is much lower than that. And then you see, again, IL-6, much lower IL-6, somewhat similar IP-10. So you're kind of benchmarking the new one, AS34, with AS01 about what it does to the immune response. But importantly, you're seeing if the concept of adsorption to alum is giving you the same favorable profile of reducing the systemic level of the drugs, which we believe will result in better tolerability safety. Next, please. And of course, well, is it an adjuvant? And that's what you see here. So this is the model antigen here. It's a GE from a herpes virus. And over to the right, you're looking at the antibody responses over dose curves to the left. After one dose, reddish. After two doses in blue. The antigen alone is not particularly potent. The molecule itself is a potent adjuvant. That's fine. We kind of expected that. But then to the right, when you absorb it onto alum, it's somewhat more potent than the adjuvant alone. But obviously, the inclusion of alum is for safety tolerability, but also for CMC advantages. And then you're seeing that, in fact, it's up there with AS01, and it's actually at the higher dose a little bit better than AS01 for antibody titers. Next, please. And probably somewhat more interesting, what is AS01 best at? Well, it's best at polyfunctional CD4 T cell responses. In this small animal mouse study, in fact, AS34 is more potent than AS01. So this is the same study you just saw, GE from a herpes virus, antigen alone to the left, obviously not particularly good at T cell response. The drug itself is somewhat better, but much better when you absorb it to alum and achieve the localization that you want to achieve in conjunction with the antigen. So you see a dose-dependent polyfunctional T cell response. And then to the right, you see what AS01 is doing with the same antigen. So you see that this AS34 is now significantly outperforming the AS01. So it's kind of beating the benchmark. So that's encouraging, even if it's just a small animal. Next, please. And yeah, I think I started off in saying that adjuvant R&D has typically been relatively slow, and we needed a lot of advance in the science to allow us to speed things up. And this is a kind of overview of how various technologies have emerged that have really allowed things to speed up significantly, we hope. And this is the kind of systems biology and being able to interrogate if the molecule is doing what you think it should be doing, if its mechanism is on target you're looking for. So this was, utilizing Ballypalendron's experience to give a kind of overview of how one might accelerate R&D around adjuvants going forward. Next, please. Yep, and I think I'm summarizing now. Hopefully, I'm more or less on time. So what's the high-level trends? Probably I've talked about this. I hope and expect that additional adjuvants will be approved in the coming years. I think we've got momentum now. You see the difference in the last decade in particular. Of course, responsible agencies will continue to ask detailed scientific questions, including mechanism of action. So if you want to bring forward novel adjuvants, great, understand how they work, be able to explain that. But, depending on the degree of novelty, it might be more of an uphill struggle than you would hope. Certainly new molecules will be discovered. Are they going to be more of the same or different? I think we've tried to highlight that there's many, many more pathways of innate activation beyond TLR that offer potential. And I've just shown you a STING agonist, and maybe some of the other speakers will talk about what they believe is going to come forward. Maybe I'm a bit biased. I tried to explain it here. Ideally, new generation adjuvants will be pure, synthetic, potent agonists, and not the typical heterogeneous natural products that we've utilized in the past. That's not to say there's any problem utilizing them. It's just in an ideal world, we wouldn't have chosen them. In an ideal world, we choose the simple synthetic. The technology was not available in the 90s and early part of this century to be able to make that choice. So we have what we have because we utilize what was available. My message is perhaps going forward, we can use easier molecules that have simpler pathways to development. And again, self-serving formulation delivery will hopefully continue to be key to success. And probably there's continued scope for seeking synergy between different molecules, as I illustrated with AS01. That was a clear path there. So thank you for your attention. I think I've got some time for questions. So please go ahead.
Yeah, thank you. Thank you, Derek. That was really good. We have one question. Would would you like to ask it, or I will?
I can see, yeah. I mean,
Yeah.
of course,
Um.
everyone is looking at AI and ML in a variety of different ways. Um, I'm probably not the person to speculate on where it's going to have the biggest impact, but for sure it's being looked at extensively in terms of readouts, in terms of molecular design, in a variety of ways. And sure, it's likely to change things, hopefully make things easier and more successful. But I'm a dinosaur. I did things the old way, so I'm waiting to learn how these tools can help us going forward.
We have another question. It says, how can we review and reimagine the maturation process 3 to ensure an adequate process for responses adapted to local vaccine production in countries such as the DRC, whose maturity level is still at 1, and while the country is experiencing a resurgence of epidemics such as Ebola and others?
Yeah, I mean, that's a complex question. I mean, arguably, the last panel we did, which was kind of how to recommend, push, make alum as readily available and able to be utilized diversely, that was a good starting point. Obviously, I'm talking about more complex adjuvants. I guess on the positive side, where's the field of adjuvants going? Well, there's kind of a lot of Me Too's. There's a kind of a lot of new adjuvants that are essentially not copies, but inspired by the originals. And there's a lot of work on trying to make them available to public sector a lot more quickly. I mean, probably IDRI was the first. IDRI's kind of disappeared now, but VFI is doing a great job in Europe of making adjuvants like the ones I talked about broadly available for the public sector. That's not my job. I work in a company. Our job is developing vaccines. And obviously, we develop vaccines for where the markets are. We have an institute in global health that does a lot too. But yeah, I think that's a challenging question. I don't have a simple answer other than I believe it's going to be better going forward because there's a lot of focus. Obviously, PATH is heavily involved, Gates is heavily involved in funding institutes to make available adjuvants for, not just the industrialized countries.
Um, Hannah, is it possible for you to unmute Didier? He has his hands up.
Yes, he's been unmuted.
Yeah. I hope
I can deal with the ones on the right, or I'll deal with whoever wants to
sure,
open
please
up
go
chat.
ahead. There's another question.
Hey, how are you?
Oh, yeah, I'm good, thanks.
Okay, Derek, thank you for your presentation. I'm Dr. Didier, pharmacist from DRC Congo. Here I am in Nairobi. I could attend in person, but I couldn't because of visa application issues. So thank you. I wanted to know what is the linkage between your presentation and the fact that no, your research and the IND initiative. I know that PAF is already in that in that fight for R&D founding development. So what is the linkage between the researches that you are doing and research and development funding? Thank you very much.
So I mean, it sounds like you're asking about a PATH initiative. Obviously, I do not work for PATH. I'm just an invited speaker. I work for GSK. My job here is just to talk about adjuvants. So I don't know about the initiatives of PATH.
Okay, perhaps colleagues from PAF can Can try to tell us what is the linkage between your work, because I think it has a linkage with research development
Um,
according
answer
according to the development of vaccines.
To answer your question, I don't think we have anything as of now immediately targeted towards, but I certainly will take this forward and discuss this with others in the team, etc., to see what can be done.
So if I just go to the questions,
Yeah, sure.
in our hands, the addition of TLR7 to the emulsion didn't do particularly well. Previously, we'd seen CPG adding to the same emulsion, doing a good job. And again, I wouldn't like to extend that beyond the actual study with HIV and glycoprotein. Unfortunately, there are not often universal observations in the adjuvant space. Formulations can work well with one antigen and not so well with another. So I wouldn't want to extend TLR7 and MF59 as a bad combination broadly. It just didn't seem to help for glycoprotein in that one study. And then the second question I see, yes, of course, we add enzymes to convert to QS21, and then, of course, those enzymes are removed in the purification process.
Okay. Thank you so much, Derek. Your presentation was just wonderful. Really, always listen to you and learn a lot more every time.
Sorry about talking about alum after you said we weren't going
No,
to. I
no,
couldn't
no,
help
not at
myself.
all. That is the starting point, of course.
Okay.
So,
Hello.
yes, is there another question?
No, it is a comment. Please, Derek, maybe a question or a comment. Please, Derek, what can be, let's say, a linkage between your work here and the ongoing Ebola outbreak in DRC and Uganda? I say, what can be involved? Your work can be involved in the now ongoing Ebola outbreak, or have you ever done a research on adjuvants that are in the formulation of Ebola virus vaccine? Have you ever done such a research?
Yeah. I mean, you're asking a difficult question about an emerging pathogenic problem that's happening right now, whereas what I'm trying to portray is 20, 30 years of adjuvant science. So my presentation today is completely unrelated to ongoing, difficult situations with Ebola right now. So I don't think there's any link.
Oh, sorry, it is about to say that Ebola vaccine formulation, do Ebola vaccine formulation, do they have adjuvants that has been done during your research, and any information you have about it?
So again, I'm not an expert in vaccines against Ebola. Of course, the last time there was a significant outbreak more than a decade ago, GSK worked hard to make a vaccine that actually worked very well. But then the pandemic, or not the pandemic, then the problem kind of went away. That vaccine was not an adjuvanted vaccine. It was an adenovirus vaccine, but it was directed against the strain that emerged then, which of course is different from the current strain. And I think the world is busy trying to understand if the vaccines developed a decade ago, including the GSK one, which has now been out licensed, are going to be effective against the currently circulating strains. I have no information on that.
Okay, good.
Okay.
I understand quite well. Thank you very much.
Yeah, thank you so much. We have a few questions which we will take at the end of the webinar this morning. We will move to Dr. Julie Joseph now. Can I? So Dr. Julie Joseph is a senior biologist and evaluator at the Health Canada Centre for Vaccines, for clinical trials and biostatistics. She has a master's in medical molecular biology from the University of Westminster and a PhD from biomedicine from the University of Würzburg, following a postdoctoral training in infectious disease and cell signaling at the University of Ottawa. Right now, she is at Health Canada, and we look forward to hear from a regulator's perspective. Go ahead, Julie.
Lakshmi, I'm just making sure that you can hear me.
Yes.
Okay, perfect.
Okay.
So
Thank you
good
so
morning,
much.
good afternoon. Oh, no worries. Thank you, Lakshmi. So good morning, good afternoon, good evening, everyone. As Lakshmi mentioned, my name is Julie Joseph. I am a senior scientific evaluator within the Biologics and Radiopharmaceutical Drugs Directorate at Health Canada. And today it is my pleasure to discuss with you the regulatory considerations for adjuvanted vaccines. Next slide, please. So before I begin, I would like to note that the views and opinions expressed in this presentation are my own, and they do not reflect the official position of Health Canada. I have, however, tried to align the views with those held by my colleagues and I within the vaccine quality divisions. Next slide, please. So when it comes to the talk outline, I have structured it in 5 different sections. I will begin with a brief overview of the adjuvant landscape from a regulatory perspective. I will then move on to discuss the current regulatory frameworks and core principles. Next, I will discuss some of the emerging regulatory themes and challenges that we notice in the adjuvanted vaccine space. I will then discuss some of the regulatory considerations when it comes to global deployment, including the need for jurisdictional alignment. And I will finally conclude the talk with some key regulatory takeaways. Next slide, please. So as I mentioned, I will begin with a brief introduction to adjuvants, focusing on the need for adjuvants and also the regulatory complexity that they introduce. Next slide, please. So in this section and later in the talk, there might be a little bit of an overlap with the topics that Dr. O'Hagan already covered, but it is an excellent talk for me to follow because some of the considerations that I would talk about would directly link back to his talk. So as we know, adjuvants are important tools when it comes to modern vaccine development. In practice, adjuvants may be co-formulated with one or more antigens, or they may be supplied as separate components to be combined with the antigen prior to administration at the point of care. From a regulatory perspective, adjuvants can add complexity to the final formulation beyond what is introduced by the antigen or the antigens. They can broaden evidentiary expectations and requirements when it comes to demonstrating the quality, the safety, and the efficacy of the final vaccine. And they can also necessitate the need for more extensive characterization, broader control strategies, and also deeper product and process understanding. So in this context, the main regulatory question is not just whether the adjuvant can improve the vaccine-induced immune responses, but also whether the final adjuvanted vaccine can be sufficiently characterized, controlled, and whether it can be supported by adequate analytical and clinical data and evidence. Next slide, please. So there are many ways in which adjuvants can add value. First, they can enhance the immunogenicity of existing vaccines. And this is particularly important when it comes to vaccines that we know have low inherent immunogenicity. And here, the examples would include subunit vaccines and vaccines containing highly purified antigens. They can also help improve vaccines that are targeting vulnerable populations. For example, the elderly, in whom we know the vaccine-induced immune responses may be lower or may be much more variable. Second, they can broaden immune responses, and this can be useful for vaccines that we know need to elicit a strong cellular immunity. And it can also be important when it comes to mucosal vaccines that require IgA responses and strong memory responses in the tissues. And lastly, they can improve formulation and delivery. So in the context of pandemics, adjuvants may be used to achieve antigen sparing, which in turn can help expand a limited vaccine supply. And they can also be used in the context of stability limited vaccines. So these are vaccines in which the antigens may not be so stable. So in this case, early formulation choices can really drive the development, the delivery, and also the deployment of these vaccines. And adjuvants may be used to stabilize the antigen and thus extend the shelf life. Next slide, please. So now I would like to discuss some of the recent developments in the adjuvant landscape. What we are seeing is a major shift from empirical design and trying to boost immune responses to trying to program immune responses. And this is something that Dr. O'Hagan elegantly described earlier. So this shift is mainly supported by a number of advances, including the emergence of rationally designed adjuvant systems. So for example, the TLR agonists and the STING agonists that are able to selectively activate the pattern recognition receptors of the innate immune system. There is an increase in combinations of adjuvants. For example, the ASO adjuvant systems from GSK. We are seeing an increase in multifunctional delivery systems. For example, the lipid nanoparticles and also Matrix-M from Novavax. There is an increase in the use of self-adjuvanted and integrated platforms. And for example, the mRNA LNP vaccines, where we know that both the mRNA and the lipid nanoparticles may have self-adjuvanting properties. And coming back to one of the questions that we got earlier, we are also seeing an increase in the use of AI and data-driven discovery tools that are being used to screen large number of candidates to identify optimal antigen adjuvant combinations and also to identify doses that might be used when going into a first-in-human clinical trial. So as you can imagine, while these developments are really pushing the adjuvant field forward, they can also increase complexity when it comes to CMC, non-clinical, and clinical evaluation. Next slide, please. So now we will move to the next section where I will discuss the current regulatory frameworks. Next slide, please. So as you might know, there are many guidance documents that are available that cover the use of adjuvants in adjuvanted vaccines. The WHO TRS 987 Annex 2 is an important reference document. There are also additional guidance documents from different regional and national regulatory authorities. For example, the EMA, the FDA, and also Health Canada. But I would like to acknowledge that not all these guidance documents cover all aspects of regulatory evaluation. Nevertheless, together, they provide a really good framework that outlines the evidentiary expectations when it comes to CMC, non-clinical, and clinical evaluation. However, several gaps and challenges have been identified in recent years. So many of these guidance documents were developed when aluminum salts were the only adjuvants around. And so there may be a disconnect between some of the recommendations in these guidance documents and the emerging adjuvant systems. There may also be differences in interpretation, which may result in differences in data expectations when it comes to the different jurisdictions. And this is something that we will talk about later. But I would like to acknowledge that despite all the challenges and the gaps, the different guidance documents are broadly aligned when it comes to general principles. Next slide, please. So there are some core regulatory principles that govern the use of adjuvants in vaccines. First, adjuvants are not licensed as standalone products. They are licensed in combination with an antigen or antigens. Second, adjuvants are not classified as active ingredients or medicinal ingredients. Third, the inclusion of the adjuvant must provide a clear clinical benefit. And lastly, each antigen adjuvant combination is a distinct product. And so the data must support each specific antigen adjuvant combination. And it is important to remember that although prior knowledge and platform data may be useful when it comes to an adjuvant, it does not remove the requirement to characterize and evaluate the final adjuvanted vaccine. Next slide, please. So now I will pivot a little and I will talk about some of the emerging regulatory trends and challenges in the adjuvanted vaccine space. And I will try to use selected case studies and examples to support these trends and challenges. Next slide, please. The first trend that we will discuss is the blurring of antigen and adjuvant boundaries. So when it comes especially to self-adjuvanting integrated platforms, the boundaries between the antigen, the adjuvant, and the delivery system are not always well defined. And this raises several important regulatory questions. For example, what constitutes the active component or the active ingredient? And also, can the components be even independently evaluated? And these questions and the answers to them become really important because they can really drive what the data expectations are. Next slide, please. So I have two case examples for this particular trend. First, we have Bexsero. As you might know, it is a meningococcal B vaccine, which contains 4 antigens, including the outer membrane vesicles. In this case, both the antigen and the intrinsic immunostimulatory activity are both embedded in the same structure of the OMV. And for Bexsero, the OMV is derived from a Neisseria meningitidis B strain, and so it is able to activate an antigen-specific immune response. As a result, the OMV was categorized as an antigen for Bexsero. And now we have the mRNA LNP vaccines, where the LNP may function primarily as a delivery system, but we know that it is also able to influence immunogenicity and also the reactogenicity of the final vaccine. So in this case, the distinction between being a delivery system and also being maybe potentially an adjuvant is less well defined compared to Bexsero. And we believe that as more and newer adjuvant systems are developed and as they evolve, this boundary and a distinction might be even less straightforward. And so we might have to rethink some of our requirements surrounding the need to classify and evaluate each of the components in an adjuvanted vaccine separately. So here, the key message is that component classification can have a huge impact when it comes to data expectations. It can influence comparability strategies, especially if the components cannot be evaluated separately. And it can also influence intellectual property considerations. Next slide, please. The second trend that we will discuss is the emergence of licensable adjuvants. So adjuvants are increasingly being developed independently from the final vaccine product. And it raises important questions. For example, who owns life cycle data and also platform knowledge? How much of that information do vaccine manufacturers have access to? And who is responsible for post-market commitments? And these questions are increasingly relevant when the adjuvant developer, the vaccine manufacturer, and the product sponsor are not the same entity. Next slide, please. So I have two separate case examples for this particular trend. First, we have the ASO adjuvant systems that was described earlier. So these are multiple proprietary adjuvant systems from GSK, and they have been used across different vaccine programs and partnerships. We also have the SWE adjuvant from CEPIC. So this is an open access query immersion adjuvant platform. And in Canada, this adjuvant has been used across several clinical programs. And when you think about licensable adjuvant, it is a powerful model because it comes with accumulated platform data and platform knowledge. However, for a new manufacturer, it is important to understand how much of that data remains accessible and how much of it can actually be referenced. So while shared adjuvant platforms may facilitate broader access, they also distribute regulatory responsibilities across multiple stakeholders. And I would like to note that having access to an adjuvant does not necessarily equate to having access to the full data package, the platform experience, and also lifecycle experience. Next slide, please. The third trend that we will discuss is an increasing trend of leveraging platform data and prior knowledge. So just as we discussed earlier, adjuvant platforms are increasingly being used across different products. And so it is important to understand how transferable the platform data and prior knowledge is, and when is it necessary to supplement the platform data with product-specific data or evidence. So one important consideration is that the selection of the antigen, the dose, the mode of administration, the formulation, and also the population can influence how much of that platform data is actually transferable. Next slide, please. So for this case study, we will use a hypothetical example of a new vaccine using a licensed adjuvant. In this case, platform data and prior knowledge may be leveraged to support the use of the adjuvant. However, regulators would require product-specific data for the antigen-adjuvant combination. So when it comes to CMC, this could include a demonstration of compatibility between the adjuvant and the antigen, characterization data for the final adjuvanted vaccine, and also stability data to support the proposed shelf life for the antigen-adjuvant combination. Sponsors would need to provide proof of concept and safety data from non-clinical studies, and they will also have to conduct clinical studies to support the safety and immunogenicity or efficacy of the product as appropriate. So while platform data and prior knowledge may be used to streamline data packages and regulatory evaluation, they would need to be supplemented with product-specific data to support the combination. Next slide, please. The next trend that we will discuss is an increasing reliance on global supply chains, and this was quite apparent during the pandemic. So these days, multiple sites across the globe are involved in manufacturing and testing the adjuvant, the drug substance, and the drug product across the product life cycle. And this level of complexity introduces quite a lot of regulatory questions. For example, how can comparability be maintained across the different sites and development stages? How should evolving assays be bridged without losing linkage to clinical experience? And how should any observed differences in critical quality attributes be evaluated and interpreted? Next slide, please. I have two case studies for this trend. For the first case study, I will focus on assay evolution and material comparability. So for adjuvanted vaccine A, the potency assay evolved throughout development. As a result, the early clinical trial lots, the later clinical trial lots, and the commercial lots were all tested with different versions of the potency assay. The sponsor also ended up using different batches of reference standards that were released using different versions of the potency assay. So the analytical bridging package that was submitted was less than ideal. So for us, the main question was if we could link the commercial lots back to the lots that were demonstrated to be safe and effective in clinical trials. So when it came to the regulatory assessment, we focused on the fact that the drug product was formulated based on protein content and not based on potency. Also, the totality of the data, including data from other clinical critical quality attributes, demonstrated that the different lots that were manufactured and tested at the different sites had comparable clinical performance. And as a result, the vaccine was licensed. So whenever there is an assay that is being changed throughout development, it is really important to generate analytical bridging data and comparability packages. In addition, a broader analytical package can help distinguish true material differences from assay-related issues. Next slide, please. For the second case study, I will focus on comparability after scale-up and how differences in critical quality attributes can be interpreted. So for adjuvanted vaccine B, commercial lots of the vaccine used adjuvant lots that were manufactured at a new site. And as a result, significantly higher particle counts were observed in the commercial lots when compared to the clinical lots. So on its own, such an observation can be quite concerning. And for us, the major question was if the higher particle counts had an impact on the safety and efficacy of the vaccine. So when it came to the regulatory assessment, we had access to data from non-clinical studies where the sponsor had compared the clinical and the commercial lots in parallel, and there was no difference in immunogenicity. We also had access to data from a small confirmatory clinical trial where the sponsor used the commercial lots, and there was no meaningful impact on safety or efficacy. And again, based on the totality of the data, the vaccine was approved. So the key message here is that differences in critical quality attributes should not be assessed in isolation, but they should be assessed in the context of the totality of the data and information, and they should always be assessed for clinical relevance. Next slide, please. We are also seeing an increasing trend to using stability data from stressed studies. So here, data from accelerated stability studies and post-degradation studies are increasingly being used to inform regulatory decisions. So it is important to understand which stability data set should drive interpretation, if the stress-induced changes are clinically meaningful, and also what the role of a post-degradation study is. And these questions are important because the different stability studies are designed to address different questions. Next slide, please. So vaccines generally do not follow a first-order degradation kinetic. And so shelf life assignment usually relies on the availability of real-time, real-condition stability data. But there are certain contexts in which data from accelerated studies or post-degradation studies can be used. So for example, an accelerated study may be used to inform interim shelf life decisions when real-time data is limited. And this is particularly relevant in the context of pandemics or early clinical development where stability data is still being generated. Post-degradation studies, on the other hand, can help characterize degradation patterns. And a comparison of degradation patterns before or after a CMC change can be used to demonstrate comparability. So stress studies can inform regulatory decisions, but they are generally used as supportive tools, and they should be interpreted within context. Next slide, please. The next trend that we will discuss is leveraging dose ranging studies that could be used to support the approval of lower antigen doses, particularly in the context of pandemics. In this case, the regulatory questions include, how can adjuvants support lower antigen doses while maintaining acceptable immunogenicity? And what level of clinical data is needed to justify an antigen sparing strategy? And a dose ranging study and an antigen sparing strategy are really useful when there is a limited supply of vaccines. Next slide, please. So I have two case examples, both of which are pandemic vaccines. So for the H1N1 flu vaccine, a phase 2 study evaluated 3 different doses of the antigen. So 3.75 microgram, 7.5 microgram, and 15 microgram with ASO3. The study also evaluated 7.5 microgram and 15 microgram, so the higher doses of the antigen without the adjuvant. Acceptable safety and immunogenicity supported the selection of the lowest dose, which is the 3.75 microgram, along with ASO3. And we have a more recent example in Nuvaxovid, which is Novavax's COVID-19 vaccine, where a phase 1/2 study evaluated 5 microgram and 25 microgram of the recombinant S protein with 50 microgram of matrix M. The study also evaluated 25 microgram of the protein without the adjuvant. And again, data supported the selection of 5 microgram of the antigen with 50 microgram of matrix M. So in both cases, the incorporation of the adjuvant was able to enable the approval of the lowest doses of the antigens that were tested in clinical trials. So lower antigen content per dose can help extend limited vaccine supply and support broader population coverage. Dose ranging studies can also be used to set enhanced specifications or smarter specifications. Next slide, please. The last trend that we will discuss in this section relates to the use of post-market studies and real-world evidence that are increasingly being used to address residual uncertainty around safety. So the important practical questions here include: when can post-market evidence help address residual uncertainty at licensure? And how can post-market data help confirm or refute early safety concerns? And real-world evidence and post-market studies can be quite powerful when sponsors are proposing to use novel adjuvants, newer platforms, or when there is a safety signal that is too rare to be adequately studied in pre-market safety studies. Next slide, please. So for this case study, I have selected HEPLISAV-B, which is an adult hepatitis B vaccine, which is a 2-dose regimen. And at the time it came for approval, it contains CpG. And so at the time of approval, the CpG adjuvant was considered novel. As a result, there was a deeper regulatory scrutiny. In addition, an imbalance in cardiovascular events was observed in one of the clinical studies. And so the regulatory review was quite complex and prolonged. Ultimately, the vaccine was licensed with a commitment to conduct a sufficiently powered post-market study. And in the end, the post-market study did not identify or confirm an increased risk of acute myocardial infarction. So this is an excellent case study to demonstrate that post-market studies can help confirm the safety of novel adjuvants or platforms where residual uncertainty remains at the time of licensure. Next slide, please. So I would like to conclude this section with a few reflections on an evolving space. So as we noticed, scientific and regulatory questions continue to evolve, and science-based regulation can help bridge some of these gaps where there is a disconnect between existing regulatory frameworks and newer adjuvant approaches. And finally, regulatory alignment can help facilitate broader use of adjuvants and adjuvanted vaccines. Next slide, please. Now I will move to the next section, which is on global deployment. In this section, we will discuss how early development and regulatory choices can influence multi-jurisdictional licensing, WHO pre-qualification, and also access in multiple jurisdictions. Next slide, please. When it comes to getting a product approved in multiple jurisdictions across the globe, there are many considerations that sponsors should keep in mind. First, it is important to understand regional requirements early. So there may be jurisdictions that have country-specific requirements, and these are easier to address early in development. For example, the sponsors may be required to generate and submit clinical trial data from indigenous populations in some cases. It is important to build a common core dossier that can be submitted to multiple jurisdictions with updates as needed. If platform data or prior knowledge is being used, it should be clearly identified and justified. And as we talked about it earlier, it should be supplemented with product-specific data as required. We recommend that sponsors try to harmonize specifications and acceptance criteria to the extent possible. This can simplify manufacturing, testing, batch release, change control, and also lifecycle management in the long run. And we also encourage sponsors to use reliance-based pathways where available. So these pathways have come to the forefront since the pandemic, And they are really useful when it comes to reducing duplication of efforts, especially if the submission has been reviewed by a stringent regulatory authority. I would, however, caution that many jurisdictions have rules and restrictions surrounding the use of reliance-based pathways. So they may have restrictions around which regulatory agency they recognize, what products can be eligible under such pathways, what the exact data requirements are, and whether reliance-based pathways can be used for the initial approval or just for post-approval changes. Nevertheless, early alignment can reduce downstream delays and also support global access. Next slide, please. There are also many challenges when it comes to achieving multi-jurisdictional alignment, and these originate mainly from the differences in how different jurisdictions manage submissions and also approvals. So first, there may be differences in country-specific documentation and requirements. And this is something that in Canada, we are also guilty of. We do have several documentation requirements that are specific to Canada. For example, we do have a document called Certified Product Information Document, or the CPID, which summarizes Module 3 information. And this is mainly procedural at this point in time, but it also can have an impact on review timelines. We also require sponsors to provide all labeling information in both English and in French. Second, there may be differences in interpretation of analytical and clinical data. So even if a sponsor submits a common core dossier, different regulators may focus on different areas of the submission and end up asking completely different questions. There may also be differences when it comes to risk tolerance, and this could originate from differences in public health needs, differences in the availability of alternate vaccines, and also regulators' experience or lack of experience with a particular adjuvant or a platform. So if you come with a common evidence package, it might be accepted as is in one jurisdiction, but another jurisdiction might ask for additional justification and even additional data. So because of these differences, sponsors often end up with divergent specifications and also additional data and bridging requirements. And this can complicate both the initial approval as well as lifecycle management. So there is an opportunity for early alignment, especially when it comes to specifications on clinically meaningful ranges. Next slide, please. So now we will discuss WHO pre-qualification, which is another pathway that sponsors can use to make their vaccines accessible in low and middle income countries. In this case, a vaccine can have an acceptable safety, efficacy, and quality profile, but it may still not meet the WHO pre-qualification requirements if the product characteristics do not align with the immunization program needs. And this is evaluated against mandatory, critical, and preferred characteristics. In addition to all the considerations that we discussed earlier, when it comes to WHO pre-qualification, there is additional focus around a number of areas. The first one is programmatic suitability. So if and how the vaccine will fit within the current immunization schedule and the delivery context. Thermostability is another important consideration. For example, there is a preference for vaccines that can be stored at 2 to 8 degrees Celsius for extended periods of time. And there is also a preference for vaccines that can withstand freezing damage. There are considerations surrounding presentation format and usability. For example, whether the vaccine is presented as a single dose or as a multi-dose, depending upon the context, whether it contains antimicrobial preservatives. There are preferences surrounding dose volumes that are to be administered in certain populations and requirements around delivery devices if they are being used. There is also additional focus around supply reliability and scalability. And additional information on WHO pre-qualification and the requirements can be found in the document that is listed at the bottom of the screen. Next slide, please. So now I will move to the last section where I will discuss some of the key regulatory takeaways. Next slide, please. So first, analytical understanding is increasingly becoming central to lifecycle management. So as products evolve through site changes, through manufacturing changes, through scale up, and also post-approval changes, analytical understanding can become the anchor that can link the product back to clinical experience. Second, platform familiarity helps, but product-specific evidence still matters. So while platform familiarity can reduce the uncertainty, it does not remove the requirement to evaluate and characterize the final adjuvanted formulation. As we discussed earlier, several factors, including the antigen, the dose, the formulation, the mode of administration, and also the population, can influence how much of that platform data remains transferable. Third, early development choices are increasingly shaping global access. So some of the choices and decisions that are made early in development, particularly when it comes to formulation and presentation, can dictate whether a vaccine is able to achieve global access, particularly when trying to go through the WHO pre-qualification pathway. And some of these decisions once made are difficult to unmake without having to submit extensive characterization and comparability data packages. And lastly, global deployment depends not only on shared data, but also on shared interpretation of that data. So just submitting a single data package may not be sufficient if the different regulators are interpreting the data differently. So early alignment, especially when it comes to data, specifications and justifications, can help ensure that the different regulators are interpreting the package in a more consistent manner. So taken together, the key message is to think early, to think globally, and to think life cycle. Next slide, please. That brings me to my acknowledgements. I would like to thank Path and the organizers for the opportunity to speak at this webinar. I would also like to thank my colleagues, Dr. Richard Siggers, Dr. Dean Smith, and Dr. Chad Owen for the many discussions on this topic. Next slide, please. Finally, I would like to thank you for your attention. I know we may have time for a few questions, but if you have questions that do not get answered in this session, please feel free to contact me at the email address displayed on the screen, and I will be happy to respond. And I will stop there. Thank you.
Thank you, Dr. Julie. Thank you so much. I think we have time for a couple of questions. One question was, both are in the chat. In case of pandemic, new pathogens, the correlates of protection will not be known. In that case, how dose pairing with adjuvant can be justified?
So going back to that question, this is something that happened with the COVID pandemic. So initially, there was no correlative protection that was identified, and it is still the case, but there was enough data coming out of all the studies that were being done, which demonstrated that the protection was somewhat linked to the generation of neutralizing antibodies. So it is not an easy question or a straightforward question to respond to. We have approved a lot of vaccines without knowing what the correlative protection is, but it always comes down to having a clear understanding of the immune pathways that are being activated and the mechanisms that we may need to achieve protection, whether it is a protection from infection or just a protection from the really severe symptoms.
The next question is there a way to characterize the research results simply to know if some of these results were obtained in laboratories located in Africa? If not, there's a prospect in this direction, given that the African continent is sufficiently engaged in the Africa CDC agenda regarding local vaccine production at the continental level.
I'm sorry, Lakshmi, did you say, is there a way to characterize?
Yes, it says, is there a way to characterize the research results? The question is in chat.
Oh, maybe I can go back to read the question because I'm not sure what characterize the research result actually means. So maybe I could go back to the question, read it, and then respond in the chat,
Okay.
if that's okay.
Okay, that's okay. Okay, we don't have any more questions at this time. Okay, there's one more. Probably we'll take it later on, um, um, after the next talk. I'll
Sounds good.
give
Thank
it
you.
over. Thank you, Julie. I'll hand this over to Robert. Take over.
Yeah, okay, great. Thank you, Lakshmi. Okay, so for our final presentation of today, we're gonna have Mark Alderson. So Mark is CSO and USI Director for MaxVax Biotechnology, which is a late-stage clinical company based in China with office in Seattle. MaxVax is developing novel vaccines and adjuvants for infectious diseases and cancer, with the mission to improve global public health. And before joining MaxVax in 2022, Dr. Alderson was a bacterial vaccine initiative leader at PATH, and he led the pneumococcal and meningococcal and Group B streptococcal vaccine programs. And then prior to PATH, he was director of immunology at GSK Biologicals in Seattle, and then before that, senior director of immunology at Corixa. So Mark, I will turn it over to you. The floor is yours.
Thank you, Robert. So let me start by thanking Robert and Lakshmi for this opportunity to talk about vaccine adjuvants for global health, and particularly, if just go back to the first slide, I want to kind of have a real focus on global health, and I'm going to focus on adjuvants, more novel adjuvants, but those that are in late stage, either licensed adjuvants or in late stage development. Let me just make a couple of comments about MaxVax. As Rob mentioned, MaxVax is based in China, but it has a small office in the US. Our vaccines are targeted towards China, but also global health. And this goes back to my global health background, working at PATH for quite a number of years. And then also the founder of MaxVax, Deshang Chen, also worked at PATH for quite a number of years before he went back to China to start MaxVax about almost exactly 10 years ago. And so we have a shared interest and history in global health. And so the vaccines that we're developing at MaxVax are targeted to both China as well as international global health. So if I could get to the second slide, I think that's the third slide. Thank you. So the way I want to structure this is to give some background about adjuvants in global health. And then when I was talking to Lakshmi about the presentation, she suggested that I talk about some of my personal history in working in adjuvants, because as Robert mentioned, quite a long time ago, I worked in the adjuvant field at Corixa Corporation, and then the corporation got acquired by GlaxoSmithKline. We had a lot of collaborations with GSK. So this goes back 20 to 30 years ago. And then I had a period of time where I didn't do much in the adjuvant field, working at PATH. It was mostly alum-based vaccines that I worked on. But then, as Robert mentioned, 4 years ago, I joined MaxVax to work on adjuvants again. And so and this is more of the novel adjuvant. So I'll tell you a little bit about what MaxVax is trying to do with our adjuvant program. So can I get the next slide, please? So just starting at a high level, I think this audience is completely familiar with the impact of vaccines on global health. That vaccines have helped to cause a dramatic reduction in child mortality since the early 1900s. But despite these vaccines, and many of which do contain adjuvants, infectious diseases remain the leading cause of childhood death worldwide. So there's plenty of work to do. And I say the easy vaccines are done, but by that, what I mean is where you don't need specific, for example, cell-mediated immune responses to provide protective immunity. So where you can take a virus and attenuate it, or you can kill a bacteria, or you can identify a key protective protein that relies on an antibody response to be protective, and alum adjuvants are sufficient. There are obviously more challenging pathogens such as TB, HIV, and malaria that do require specialized adjuvants. These were touched on a little bit earlier. I want to go into this in a little bit more detail. Or it can be delivery systems in order to promote protective immunity, which is often cell-mediated immune responses. Next slide, please. So just looking at the leading causes of death, this is in low-income countries, and you can see, and this is not just childhood, this is overall. So you'll see things like ischemic heart disease and stroke. But when you look at the infectious causes, lower respiratory tract infections, number one, this is obviously caused by a number of pathogens, some viruses, including RSV, influenza, COVID, etc., other viruses, but also bacteria such as pneumococcus. We have diarrheal diseases, and then malaria is obviously a major factor. We have, as Derek mentioned, 2 recently licensed vaccines to prevent malaria. We come down to TB. We have not had a licensed vaccine for TB for more than 100 years now. And so these particular pathogens do cause a lot of mortality, particularly in low-income country settings. Can I get the next slide, please? So why focus on novel adjuvants as opposed to alum-based adjuvants? So the traditional adjuvants, like the alum-based adjuvants, are insufficient for eliciting protective immunity to certain pathogens. So I would agree with comments that Derek made. If alum is sufficient, you should go with alum, but in some cases it's not. And certain pathogens such as malaria and TB clearly require more potent adjuvants and adjuvants that are steering the immune response more towards cell-mediated immunity. In addition, novel adjuvants are often needed for individuals with suboptimal immunity, such as older adults and immunocompromised individuals. This is an area of focus at MaxVax as well. And then, as we heard in previous presentations, particularly from Derek, about the potential to reduce antigen dose or the number of doses administered. And I think the hepatitis B vaccine is a good example of that, as shown in Derek's presentation. By adding in a more potent adjuvant than alum, you can reduce the number of doses to get full seroprotection against hepatitis B. Next slide, please. So challenges for developing novel adjuvants for global health. Certainly, some of these have been already discussed, so I won't go into some of those in detail, but one of the things I did want to talk about is translation from preclinical to clinical. The saying goes, mice lie and monkeys exaggerate. And so as I think the kind of studies that Derek was talking about, looking at different adjuvants in a clinical setting are absolutely key, because you will see different results in preclinical studies compared to a clinical setting for a number of reasons. I think, having spent a lot of time working on conjugate vaccines, that's probably a good example. So conjugate vaccines for pneumococcus, meningococcus, and group B strep. What you see in mice is that there's an absolute requirement for an adjuvant, i.e. alum-based adjuvant, to get a decent immune response in mice. As you go up to rabbits, you see a better response from the conjugate alone, but still enhanced by adding in the adjuvant. You get into monkeys, and you'll see a good response, slightly enhanced by the adjuvant. But typically in the clinic, what you see is very little of any boosting of the immune response to conjugate vaccines. And oftentimes, like in the case of pneumococcal vaccines, the adjuvant is there more to stabilize the antigen, as has been discussed by previous speakers. One of the big issues, of course, for global health is manufacturing and supply. We touched on this a little bit, but having the ability to manufacture large numbers of doses of adjuvants where requirements for the vaccine are going to be in the millions to tens of millions of doses becomes a key issue. And this is an area that MaxVax has focused on, so I'll get to that more in a moment. There are intellectual property issues when it comes to global health. This was also touched on previously. Limited clinical data. So what I mean by that is that there are many adjuvants, novel adjuvants that have made it into early clinical evaluation, but have not progressed beyond phase 1 or phase 2 studies. So as we heard previously, the number of licensed adjuvants or licensed vaccines that contain novel adjuvants is relatively small. And part of that is building up that clinical database. Regulatory, I'm not going to touch on that. Julie went through this in extensive detail in the previous presentation. I did want to touch just a little bit on the safety tolerability. Derek mentioned this, but oftentimes what happens is as you enhance the immune response promoted by your adjuvant, the stronger the adjuvant, the more reactogenicity you get. There's often a correlation between reactogenicity and the immune response that's elicited. So part of the key there is trying to get the right balance of enhancing immune responses, but also making sure that the adjuvant has a good safety profile and good tolerability profile. And then finally, the cost. This was also touched upon when you get to very complex adjuvant systems like AS01. You have multiple components. The QC testing, analytical testing is very complex. These are expensive adjuvants, complex adjuvants to manufacture, which makes it challenging to keep the cost of making the vaccines and therefore the price down to reasonable levels from a global health perspective. Next slide, please. So I just wanted to put in this as a reminder to for the overall complexity of vaccine development and the time involved in doing this. So this is just a summary of what is involved for vaccine development, from target validation, preclinical through early stage clinical, phase 2, phase 3. And this is a very, very long process, particularly in the context of global health. And when I was putting this presentation together, two of the examples that I really thought about were the malaria vaccine that's been mentioned previously, RTSS, which took approximately 35 years from its initiation to when it actually got licensed and put into use as a vaccine against malaria. The other one is TB. I'm going to talk about some of my early experiences working with a TB vaccine, and that work was initiated roughly 30 years ago, and the lead candidate now is in phase 3 trials, of course, which I'll touch on. So very complex process, but when you include novel adjuvants, then it adds another layer of complexity to the development of vaccines. Next slide, please. So just, I won't go through this very quickly because this has been discussed by previous speakers, but there is a limited array of adjuvants that have been licensed. The aluminum, of course, as we discussed, was licensed back in 1930s. And then over the since the 1990s, 2000s, there have been a number of other adjuvants. These all got mentioned in previous presentations, so I'm not going to go through them in detail, but just to reiterate about the combination adjuvant systems. We're taking combinations of delivery systems such as liposomes or all-in-water emulsions, and then adding in various immunostimulants such as TLR agonists and QS21, which has been discussed extensively. Next slide, please. So I did want to mention a little bit about AS01 and my personal experience with AS01, but just by way of summary, since Derek went through this quite extensively, the AS01 contains a combination of two immunostimulants, MPL, which, as was pointed out, is purified from bacteria. So this is a complex manufacturing process. QS21, which is purified from the bark of the Quillaria saponaria trees, which is also a complex manufacturing, and it is a mixture of molecules in a liposomal formulation. But in recent years, this adjuvant system has been licensed in a number of vaccines, as we heard, 2015 for RTSS with AS01E. There is, of course, a more recently licensed malaria vaccine, R21 Matrix M. The shingles vaccine, which contains AS01B. This is the higher dose of the adjuvant. So AS01B contains 50 micrograms of MPL and 50 micrograms of QS21, and the AS01E contains half those contents for the two immunostimulants. So this was licensed in 2017. We heard about Arexvy, which is licensed in 2023. And then I did want to talk a little bit about the TB vaccine that is in late stage development, M72, which is now in phase 3 and contains AS01E. Can I get the next slide, please? So this is a picture from one of the review articles from GSK. Derek showed this slide as well, just describing the two molecules that are in AS01, how they act differentially from each other, but they act in concert to promote strong immune responses. So the toll-like receptor 4 molecule that's expressed on the cell surface, that is the target of MPL, and then QS21, which is taken up with inside lysosomes and forms the inflammasome, and you see promotion of pro-inflammatory cytokines and enhanced antigen presentation, which leads to both enhanced B cell responses and enhanced T cell responses, which what is likely to be key for, I believe, for the TB vaccine. Next slide, please. So this goes back to some of the work I was involved with in the late 1990s, early 2000s on TB. So as I was working at Corixa Corporation, we had a collaboration with GSK on developing the TB vaccine. And the lead candidate that came out of this work, we referred to as MTB72F. It's now referred to as M72. It's been altered slightly. The construct has been altered slightly. But one of the things we did in the early days is because of the collaboration with GSK, we got to assess a number of the adjuvant systems that GSK was working on, including AS01 and AS02, which is no longer really one of the lead adjuvants from GSK. What I wanted to point to is on the right-hand side of this slide. This is from a publication from the Corixa GSK groups more than 20 years ago, is the testing of the AS02 and versus AS01 adjuvant. So just to describe the difference here, both of these adjuvant systems contain MPL and QS21. The AS02 is an oil-in-water emulsion, and AS01B, as we've heard many times, is a liposomal formulation. So in this particular system, this is data from mice. We have on the left-hand side is antibody data. In the middle, it's gamma interferon secretion. and on the right, gamma interferon ELISpot data. So gamma interferon is thought to be a key component in the protective immune response against TB. And if you look at spleen cells from mice that were immunized with M72 with ASO2 versus ASO1B, for the ASO2, you can see that if you look at the most immunogenic component of this fusion protein is MTB39, you can see a good response. Each spot here represents a T cell that's making gamma interferon, and you can see the spots with ASO2. But when you go to ASO1, there are so many T cells producing gamma interferon that you can't see the spots anymore. It's just blown out the wells with complete color. So this was part of the work that led to us moving forward. Actually, both adjuvants were assessed clinically, but based upon superior T cell responses with the ASO1B adjuvant. This was the adjuvant that moved forward into later stage clinical trials. Can I get the next slide, please? So this is data from M72 ASO1E. So the decision was made to go with the lower dose adjuvant ASO1E. This is phase 2B data that was published in 2019. And it's showing that in individuals latently infected with MTB, that the vaccine could reduce the number of subjects that progress to active TB. So this is the placebo here. This is the antigen, the M72 of ASO1E vaccinated subjects, and there's about a 50% reduction in the number of subjects that progresses to active TB. So, 50% is not ideal. Clearly, you would like to see higher percentage than this, but this, given the burden of disease with TB, and given that there has not been a new vaccine license for TB for more than 100 years now, this was very encouraging data. Based upon this, if I can get the next slide, this candidate, M72 ASO1E, was progressed to a phase 3 clinical trial. Shown here, this began a couple of years ago. It was fully recruited, 20,000 subjects, a little over a year ago. It is an extensive study, 54 clinical trials, sites across 5 countries in Africa and Asia, and the interim results are expected in 2027. So we'll be very exciting to see those results, but just as a reminder, this particular vaccine has now been in development for almost 30 years. Can I get the next slide, please? So I want to shift gears now and tell you a little bit more about what we're doing at MaxVax with respect to novel adjuvants. So we have a number of technology platforms, but really the main focus of the company is developing recombinant protein vaccines, many of which contain novel adjuvants. Some contain alum-based adjuvants, but our focus really is on the novel adjuvants. So this can be prophylactic vaccines for human infections, therapeutic vaccines. By that, I mean individuals that are already infected with the organism. So in the case of, for example, chronic viral infections or TB, for example, and we are dabbling a little bit in mRNA technology, but again, the focus is really on the adjuvant side. Next slide, please. So I think one of the things that distinguishes MaxVax from a lot of smaller up-and-coming biotechs, as a reminder, we've been going for about 10 years, is our manufacturing capabilities to manufacture adjuvants and vaccines at large scale. So we have a number of facilities. The first facility has been operational since 2020. This is based in Chengdu in China. And this is where we manufactured adjuvants and vaccines for phase 1 and 2 studies, currently ongoing in China. And then 2023, we opened an additional facility. This is commercial scale for making material for phase 3 and for commercialization of our initial vaccines. And then we have under construction an additional facility, which is adjacent to this facility, also in Shanghai, that will increase our capacity for manufacturing of both adjuvants and vaccines. Next slide, please. Just quickly, our pipeline of vaccines. I'm not going to go through this in detail, but we have progressed our most advanced vaccine candidate, which is vaccine for shingles. So similar to Shingrix, as described by Derek earlier, we have taken this candidate, and I'll tell you a little bit more about it in a moment, through a phase 3 clinical trial. Based upon the interim results from that phase 3 trial, we submitted a licensing application in China in October of last year, and we expect to hear back on that application by the end of this year. So this is our most advanced program. It does have one of our novel adjuvants in it. We also have an RSV vaccine for older adults that is in late stage development. So that's currently in a phase 3 efficacy trial, also in China. We do have plans to try and market these products outside of China eventually. We have a rotavirus subunit vaccine that's in phase 2. This is alum-based. And then I won't go through the others, but RSV maternal is our final one that's actually in the clinic. It is an RSV pre-F based vaccine, but it does not include an adjuvant for the maternal indication. And just maybe quickly, the HPV therapeutic, TB therapeutic, these are both candidates that are preclinical, but we have our advanced adjuvant systems included in those candidates. Next slide, please. So the goal of our program is to develop adjuvant systems that are scalable from a manufacturing perspective and are tailored to the protective immune response required for the particular pathogen of interest, but also that these adjuvants would be well tolerated and affordable. So vaccines for certain viral diseases, we talked about this, RSV, intracellular bacteria, parasites, and vaccines for cancers. We also discussed a little bit, shingles vaccine obviously is a vaccine for older adults. The RSV vaccine is also for older adults with declining immune systems. And the poster child for this, of course, is GSK's AS01 adjuvant used in Shingrix and Arexvy for older adults. One of the things that we have been one of our goals is to also find partners for our adjuvants. Since we have been developing expertise in adjuvants and formulations, this is something that many vaccine developers that do not focus on the adjuvants don't have the knowledge and expertise on adjuvants and formulations, access to the raw materials that are used to make these adjuvants, being able to make the adjuvants at commercial scale, and the financial and time costs associated with assessing and accessing adjuvants. So this is, we have efforts to try and partner our adjuvant programs with groups that are interested in having access to adjuvants, but don't currently have access or don't have the expertise to develop their own adjuvants. Next slide, please. So a little bit more about our adjuvant program. So we have designed and manufactured over 20 adjuvant molecules. Most of these are synthetic. We have worked on a number of delivery systems that have already been discussed, liposomal, oil and water emulsions, aluminum hydroxide. We have developed a number of adjuvant formulations for preclinical research testing. And then from the standpoint of moving products forward, we've scaled up production under GMP for 3 of our adjuvant molecules, 2 lipids, 2 delivery systems, and 5 formulations that are currently produced in our pilot or commercial manufacturing plants under GMP compliance, with the goal of making these scalable for global health applications. So we have applications, as I mentioned, to both our in-house programs and potential partner vaccine projects. As I mentioned, one adjuvant for our shingles vaccine, which I'll tell you about more in a moment, is at the licensure stage. We have a different adjuvant that we're using for our RSV vaccine that's in phase 3, and a number of others that are in preclinical development. Next slide, please. So these are some of the molecules to make our adjuvant formulations that we are currently manufacturing. So some of these will sound familiar since we've heard a lot about this in previous presentations. QS21, which is purified from bark. As we heard previously, we source the raw materials and do our own purification. Our current batch size is about 1 million doses. We have been working on a TLR3 agonist, poly-IC, that we can make at large scale in-house at MaxVax. We have, and that is in clinical assessment. We've also been working on TLR4 agonists, that's currently preclinical. And then some of the lipids to use to make our formulations. So we heard about DOPC that's used in AS01 and other adjuvants. We manufacture that internally at MaxVax, as well as a DOTAP as an alternative lipid for making liposomes, cationic liposomes, and then other molecules that are more in the research phase. But like was mentioned by Derek, we are also looking at making semi-synthetic QS21 in a similar manner to what was described previously. Next slide, please. So we've talked about QS21. I'm not going to go through this in detail because of that. We have developed our own purification process for this. Just to reiterate that it is a mixture of these molecules here, so it does make manufacturing challenging to make sure you have a highly reproducible process and well-characterized adjuvant or molecule QS21 or mixture of molecules. So we have developed this process, as I mentioned. It is being used in actually all of our lead vaccine adjuvants that I'll tell you about more in a little bit more detail: MA103, MA105, and MA107. And MA103 and MA105 are both in phase 3 clinical development. So key for us is QS21. Next slide, please. We've been using poly-IC as a second stimulus instead of TLR4. We manufacture this internally at MaxVax under GMP compliance. This is the structure, so it's a kind of an analog of double-stranded RNA that triggers through toll-like receptor 3. As a standalone, part of the reason we chose to work on poly-IC is because in China, poly-IC is a licensed product. It is licensed for treating shingles and chronic hepatitis B. And it has been fairly extensively studied as a vaccine adjuvant in early stage clinical trials, but is not licensed as an adjuvant as yet. So we have made this at kilogram scale, and it is in our lead adjuvant system called MA105, which again, that's our shingles vaccine candidate adjuvant, which I'll describe a little bit more in just a minute. Next slide, please. We've also been working on synthetic TLR4 agonists or synthetic MPLs. We've worked on a number of molecules. This has not made it into the clinic yet, although we have ongoing efforts trying to define our lead molecule. And the idea is that we would combine this with QS21 in a liposomal formulation, so somewhat analogous to ASO1E. But I did want to emphasize the advantage here of having a synthetic MPL because it can be a single structure as shown here, whereas the MPL that's currently used in ASO1 is, as we've discussed before, is purified from bacteria. So it is a mixture of congeners. So it is challenging from a manufacturing standpoint and challenging from a characterization standpoint when you've got mixtures of different lipid molecules. Next slide, please. And then finally, the delivery systems that I mentioned previously. So we have alum that we can make at large quantities, oil and water emulsions, and our liposomal formulations, which are the basis of our lead novel adjuvants. Next slide, please. Okay, so these are our three lead adjuvants. I mentioned them a couple of times now: MA103, MA105, MA107. I will go through these in a little bit more detail. So the MA103 is in our RSV vaccine, MA105 is in a shingles vaccine, and also in our therapeutic HPV vaccine. I'm not going to talk more about that, but that is also targeted for global health. And then more recently, we've been developing MA107. So this is the TLR4 plus QS21. So most like ASO1. And there we're looking for partnering for global health indications such as malaria and TB. And we do have an internal, as I mentioned, TB program, but it is very early stage. Next slide. Okay, so MA105, again, this is our lead adjuvant because it's in our shingles vaccine. It's also in our therapeutic HPV vaccine. As a reminder, it contains poly-IC and QS21 in a liposomal formulation. We present this particular vaccine in a prefilled syringe, so there's no reconstitution required. And the goal here of this program for shingles is to promote similar immune responses to ASO1, i.e. in Shingrix, but with lower reactogenicity. So this is one of the issues with ASO1B, the high dose formulation. It does have causes a relatively high frequency of local and systemic adverse events. And particularly when it gets to grade 3 adverse events, according to the US CDC, it elicits about 17% of grade 3 adverse events. So our goal here is to try and get a vaccine with our adjuvant that's similar to ASO1 or similar to Shingrix, but with better tolerability. And unfortunately, I can't show you any of the data from our phase 2 and phase 3 trials as it's not published yet, but let me just say that in phase 2, we did a head-to-head study compared to Shingrix in subjects in China, and we had 8-fold less grade 3 adverse events with our vaccine compared to Shingrix. And then we recently got our interim phase 3 data, and we had a internal target of vaccine efficacy against shingles of at least 90%, and we hit our target there. So based upon that data, we have moved forward with the licensure application that I mentioned in October of last year. So now with this particular adjuvant having gone through phase 3 assessment, we now have a safety database of around about 13,000 subjects. Next slide, please. So MA103 is our next in line. This is the one that we're using for our RSV vaccine candidate for older adults. This one contains QS21 in a liposomal formulation, so it's a simpler vaccine formulation compared to MA105. It's also presented in a prefilled syringe. The goal here for this particular program for RSV is to get a vaccine that has similar efficacy to the licensed protein vaccines from GSK, Arexvy, and Pfizer with Abrysvo. And as I mentioned, it's currently in a phase 3 clinical trial in China, so 25,000 subjects, with the indication being the primary endpoint being RSV, lower respiratory tract infections. So we'll get the data on that trial about mid-year, so in the next few months, to make a decision about moving forward with a licensure application. Next slide. And then MA107, this is preclinical, but as mentioned, this is our lead adjuvant system for malaria and TB. Also can be used for other vaccines, of course. Goal is to have similar adjuvant properties to AS01. The commercial scale process is under development, and we'll leverage our existing experience in making QS21 and liposomal adjuvants, i.e. MA103 and MA105, from the standpoint of developing a commercial scale process. Next slide. So I wanted to show you just a little bit of preclinical data with some of the adjuvant formulations that I've been talking about. This is a piece of data where we compared MA105 and MA107 in a preclinical model in mice with the GE antigen from VZV, the shingles vaccine candidate. And on the left-hand side, showing serum IgG2A responses. So this is an indicator of a gamma interferon Th1 type response. And if you look at the controls here, what you can see is you get a nice boost in the IgG2A response with either an adjuvant that this one is essentially MA105, and then we see over here, this is MA107. This is an alum-based with the same immunostimulants. It's a slightly lower immune response, but MA105, MA107 are about the same. For T cell responses, which is thought to be key for protection against shingles, what we see is the negative control here. This is MA105 and MA107 here, and again, compared to an alum-based control. So these are T cells expressing IL-2 or interferon gamma, or the combination of the two in a preclinical model. Next slide, please. This is a second study, also in the GE system, just looking at MA107 on its own compared to a alum adjuvant, looking at subtypes of antibody. And as you can see, what the alum does is boost the IgG or the IgG1 response to GE quite nicely, almost as good as what you see with MA107, but boosting the IgG2A, G2B response, gamma interferon dependent, you really only see boosting with the MA107 adjuvant. Next slide, please. This is going to a different model system that we've used quite a bit. That's RSV, using a pre-F protein. That's part of our vaccine candidate, showing again preclinical studies in mice with the MA107 adjuvant here. So 902 is the name of the antigen here. These are the IgG responses. So you get a fairly good response with the antigen alone. It is boosted somewhat with MA107. And you do see a boost in G1 and G2A responses, not so much on G2B. the key for RSV is the induction of a neutralizing antibody response, and that's what's shown on the right-hand side here. And you can see the 902, the antigen alone, gives a modest response, a titer of around about 150, whereas if you add in MA107 to boost this response, it goes up almost 2 logs increase in the neutralizing antibody response. So this is, again, showing the benefit of adding in a potent adjuvant such as MA107. Next slide, please. I think this is the final data slide I have. This is showing the impact on the T cell response for the pre-F protein. So one thing that MA107 or MA105 does, and MA103 as well, is induce very nice T cell responses. So on the left-hand side, we have CD4 T cell responses with various cytokines. You can see the antigen alone really does nothing. It gets boosted nicely with the adjuvant. And on the right-hand side, these are CD8 T cell responses. So in the mouse systems, you tend to see this, that these adjuvants will boost CD8 T cell responses. It's unclear yet whether these adjuvants will be sufficient to induce CD8 T cell responses in the clinic, but in this particular case for RSV, it's felt like CD8s are probably not essential for protective immunity. Next slide, please. Okay, so just to summarize what I've been talking about, so novel and potent adjuvants are required to develop vaccines against challenging diseases such as malaria and TB, where classical adjuvants have been unsuccessful. And I think what has become clear is that multi-component adjuvant systems such as AS01 have been recently incorporated into a number of licensed vaccines, malaria, shingles, RSV, and in late stage development, such as TB. And our role here in developing vaccines and adjuvants is really to make sure that we can manufacture these at high quantities, high quality, and low cost that are suitable for global health applications such as next generation malaria and TB vaccines. And to date, more than 25,000 adult subjects have been immunized with vaccines containing these novel MaxVax adjuvant systems. And I think that is it. Next slide. Just thank you for your attention and happy to take any questions that may come through.
Great. Thank you very much, Mark. That was excellent. Really, really fantastic presentation and great to see all the work that you've done at MaxVax. So we have just about 12 or 13 minutes left in our time today, and we have a few questions that have come through on the chat. And so I will try to get to as many of these as I can. Let's see. I think the first one, maybe going back to Derek. There was one more question on yours, on your presentation, regarding TLR4 and TLR7 agonists. So basically the question is, with the potential for of SMIPs and TLRs, what do you think about how exploratory studies focused on TLRs could reshape the adjuvant field? So Derek, if you wouldn't mind commenting a little further on those two, that would be fantastic. Thank you.
Yeah, I mean, if you want to reshape the adjuvant field, maybe move beyond TLR agonists. So there are many additional receptors. I focused on STING. Probably tomorrow there'll be more alternative approaches being promoted. I think maybe we've exhausted TLR agonists. We've used them extensively. Maybe there's different molecules we can focus on.
Okay, great. Thank you for that. Okay, and then there's a few questions for Julie. So Julie, the first one is, I think I wanted to go back to the question that I think was mentioned earlier regarding characterizing research results, because there was a question about basically about assays, results were obtained from laboratories in Africa. And basically, if you could comment kind of more generally on how regulatory agencies are viewing the use of assays that are done in endemic settings. I think the question the participant was asking about the Africa CDC agenda regarding enhancing local vaccine production capacity. So if you have thoughts along those lines, it'd be great to hear them. Thank you.
So I'm not sure I understand the question clearly, but based on what I think it is, when it comes to evaluating analytical data and also clinical data from different jurisdictions, there are requirements surrounding the need to have these sites and assays being done under GMP and GLP requirements. So that in itself, I believe, would exclude data coming out of research facilities and such circumstances. And this is when it comes to the approval of the product. So the final licensing.
Okay, great. Yeah, thank you for that. I think that makes sense. Okay, and then Julie, one other question for you was on the your perspective on the potential for standalone regulatory evaluation or approval pathways of adjuvants. And I know we touched on this a little bit, but the participant was asking about if there's anything analogous to the emerging novel excipient initiatives that are being explored by the US FDA, and considering that adjuvants are not inert not inert components in the sense of conventional excipients and can have their own independent effects. So maybe if you could say a little bit more about the potential for kind of standalone pathways and how that might work.
So I do agree that adjuvants are not excipients per se, because they may have their own immune modulating effects. But it is also important to remember that the adjuvant is unable to induce an antigen-specific immune response unless it is combined with that antigen in question. So when it comes down to it, I do understand the attractiveness of having independent approval pathways for adjuvants, but they are not considered drugs that have an independent therapeutic or prophylactic indication. They are still components of an adjuvanted vaccine. So I do not believe that there is an opening for adjuvants to be approved separately and independently. What we have instead is the ability for sponsors to submit drug master files, which can then be cross-referenced by the independent vaccine manufacturers. And such a change would require quite a lot of regulatory will across the different jurisdictions. And even if we go down that path, it's going to take years and years.
Okay, great. Yeah, thank you for that. I think that's clear. And then maybe one more question, Mark, I want to bring you in, and there's a question on basically on mucosal adjuvants and the adjuvants which are specifically inducing mucosal immunity. And I mean, maybe this is from the presentations or the areas which you mentioned, I think maybe is relevant for TB vaccines and adjuvants particularly, but I was just wondering if you could comment on, what you see as the potential for mucosal adjuvants and how those might fit in with some of the other programs or other programs you have at MaxVax.
Sure. Thanks for that question. So I think the way I think about it is, from the standpoint of mucosal delivery of mucosal delivery of vaccines, I think the way I look at it is if you can develop a vaccine that does not need to be delivered by a mucosal route, then that's a plus. That's the way you should go, and you really should only think about that if you can't get sufficient protection from a systemically delivered vaccine. So that may be the case, for example, in something like TB, where as I showed you with M72 AS01E, you are seeing about a 50% protection. The malaria vaccines clearly are not optimal either. So there may be issues around there, but it is challenging to develop adjuvants for mucosal delivery, particularly if you're thinking about kind of intranasal administration. So it does increase the complexity. It's not something we're focused on right now at MaxVax because we are following in kind of in some ways a well-trodden pathway for some of the vaccines that we've been working on. Obviously, the shingles vaccine that we're developing, GSK has demonstrated with Shingrix that you can get well above 90% vaccine efficacy. The RSV vaccines so far have also looked pretty good, but I think there are certainly some areas where mucosal delivery is absolutely worth looking at, whether the adjuvants that we're working on are going to be the best for eliciting mucosal immune response. I think that is still to be determined. There are clearly adjuvants that are more targeted towards trying to promote mucosal immune responses, such as the DMLT that was being worked on at Path for a number of years. We're not currently doing that at the moment. So, but I do think there is scope for mucosal delivery of vaccines, but only when it's absolutely necessary. I think the preferred route is to go the systemic, and it's only really if you can't get a sufficient protection from systemic delivery that you would want to go down the mucosal route.
Okay, great. Yeah, thank you for that. And Derek, we just have a couple minutes, but maybe if you wouldn't mind weighing in on the mucosal immunity question. And I mean, in particular, I'm wondering, are there for the adjuvant systems from GSK that you talked about, is there potential for using those in mucosal delivery context, or do you think we need like basically a new class or separate class, different types of adjuvants for enhancing mucosal responses?
Almost certainly. So the adjuvant systems were developed for injection, so there's no reason to expect that they would be successful mucosally. There are different challenges mucosally, of course, different tissue barriers to think about, and different mucosal routes bring different challenges. Obviously, oral is the most difficult. So if you want to develop a mucosal adjuvant, you need a distinct and separate approach involving molecules, formulations appropriate for the route. It's been evaluated for 30, 40 years that I've been active, and there is no single mucosal adjuvant system that's yet emerged, which really implies quite how difficult it is. But even if you step away, the bigger problem is we don't know how to license mucosal vaccines unless they do the same thing as injectable vaccines. So immune correlates often not there. Efficacy trials necessary, difficult, challenging, complex. So there are many challenges for delivery of mucosal vaccines beyond mucosal adjuvants, but we don't have broadly applicable mucosal adjuvant systems. But some of the molecules we use may be useful, and other molecules may be more useful, like DMLT that was mentioned.
Great. Okay, thank you for that, Derek. Okay, we're just about at time, everyone. So just like take one more chance to thank all of the speakers for today. Greatly appreciate all those presentations. And we just a reminder, we will continue tomorrow at the same time. We have 4 more presentations, as you can see on the slide there tomorrow. And so we're looking forward to hearing those and getting some feedback from all the participants. And then just lastly to mention, there was one comment or question about the survey. We will send a survey for feedback after the presentations tomorrow. So I encourage all the participants to look out for that. And with that, I think we can close for today, and we'll look forward to speaking with everyone again tomorrow. Okay, thank you, everyone.